Good morning.
Good morning. How are you?
Okay. I apologize if things were a little chaotic yesterday. Each year we try out some new lectures and some work and some don't. So we appreciate your patience.
Yeah, I just used the time to go back to one of the other lectures that I got behind on and worked on that. So that was no problem. So
Okay.
I appreciate all the instruction y'all are giving. It's really helping me. So
Well, again, it's a team effort, and we do learn. I mean, but the reason we do those polls about the modules is that we take them seriously and we do revise them. So unfortunately, it won't help you directly, but for the next year's group, we do revise the lectures each year carefully.
yeah, I can tell y'all really work hard on it to make it accessible for everyone. So I appreciate it.
I mean, the other thing that's difficult is we have experts and we also have people who've never done any computing. And so we try to balance and I see there are a couple of people actually are coming for the second time.
Oh, okay.
They took it last year, and I guess I
Yeah,
always have said that if something's worth hearing once, it's worth hearing twice.
yeah.
If it's worth hearing at all, it's worth hearing more than once.
Well, I have a PhD in biology, but I've never really coded. But I just joined Michael Sachs's lab during the shutdown, and I'm kind of the resident biologist, and everybody else does the modeling. So I'm trying to get up to speed on, some of the programming and modeling stuff so that I can.
Well, I would definitely say that, I mean, my students get tired of hearing this stuff. I always say the same thing, which is the biology is the hard part.
Oh,
You understand the biology, well, converting it to computer code is actually pretty straightforward.
okay. Yeah.
Now, sometimes there's a gap because what we understand biologically isn't a full enough picture to be able to build a model.
Mm-hmm. Yeah.
And another slogan I have is that building a model, very often when you build the discipline of building the model tells you what experiments you need to do because you realize there's missing information.
Uh-huh.
Building a model is brutal about telling you what you don't know.
That's for sure. Yeah. I'm reading a book on information theory just for these reasons, because we have, this piece of data and this piece of data, and you put it together and you're like, oh, we're missing all this other stuff, So information theory is really good at helping you figure out what parts you really need to focus on getting so you can actually make that model.
Yeah, well, there are so many disciplines that you need, and nobody, I think, comes to this course knowing everything they need. At least in my experience.
To learn, right?
Right. That's one reason we do the hackathons, because that teaming can be very productive for people. Not always. I mean, some people it doesn't work for, but for a lot of people, it's quite useful. And I apologize. I'm gonna have to go just set up a little bit. I'm getting some emails from people.
I need to download the slides and stuff.
Okay.
So
Yeah, and so we'll be, if you want to download the exercises, so we're starting with the lecture that I should have done yesterday, which will be 3.4. So it'll still be numbered 3.4, even though it's in module 4.1. And there's also a demo, which if you want to download that and upload that either to NanoHub or to your local copy of whatever you're using, that would be good because we'll be using that in the morning. And I hope I'm sorry we don't have more time when we do this in person. I have time to meet everybody and chat.
Yeah.
Now it winds up, I'm the one who has to run the Zoom session, so I don't get to do as much. But I'm glad we got to talk a little bit. I'll be back and I'm happy to talk more if we could find the time. If you excuse me just for a few minutes, I'm going to just do some setup and I'll be so if you hear me typing, you'll know what.
Yeah, no worries. Sounds good. I'll see you in a few minutes.
Okay, great.
Hello.
Hello, Lorenzo. I'll just getting things set up here. Trying to move through all the punch list. There's so many things to do. I get sometimes get a little bit behind on the lecture list. So let me just make sure I have. How are you doing? You probably aren't getting very much sleep with all the help you're giving us here.
Yeah, well, not a big problem. I had a meeting this morning at 8, but it's okay. I'm enjoying a lot, so it's cool. After 6 months, I figure out finally how to avoid having to register every time at the Zoom meetings. I think
It's supposed to let you register. It's set up so that you should be able to register once and then select all of them.
Yeah, I learned how to do it after 6 months yesterday night. So it's an achievement.
Well, I apologize if you have to, if you're. Okay. Okay. Oops.
James, can you stop the cloud recording 1 second so I can push on my, and I can allow the local recording, otherwise it won't allow me.
It won't allow the record. Okay.
So if
That's
I
interesting.
stop 1 second and then I start, and then you should be able to start again where you were, as we did yesterday. Let me see. So if I do like this, and then I do this, record on this. Now I'm recording on my local computer.
Great.
Then I
Because
think
today there's this issue that I have to, the one thing I have that I have to do after with the IMAG meeting this afternoon.
okay.
So I have to leave, I have to run that meeting. So I'll be gone for, I don't think you're going to miss much if you're out there, but I have to be gone for those 2 hours this afternoon.
Yeah, I was thinking to stay here today. And I
but
can
it's good.
record, so no problem for that. The other session, I had the full recording, but I have to enable, now is recording, I think, on mine. So
Great. Is the screen share okay?
I'm not seeing anything at the moment, just a gray screen. I don't know the others.
I wonder what happened. Okay, well, let me see why that is the case. It should be.
Now is good.
Huh. I wonder what happened. So now you've got the screen.
Yeah, now is the usual. Good.
There was an update on Zoom the day before yesterday, which was not a helpful one. So, welcome everybody. I appreciate you being here. Appreciate people coming back. And all right. Are the closed captions visible to people?
Not at the moment. Oh, now yes.
So if people are waiting, I encourage you to go and download the slide deck that we're going to be using will be numbered 3.4 version 3. So I could put that in the, Giuliano, maybe you can also put that in the chat that we'll be using, and also the instructions for downloading the demo that we have, which is also under day 3, module 4. I need to do the breakout. No, I was forgetting. if things went a little fast yesterday, I appreciate your patience. You'll have plenty of opportunity to go back over that material again. Thank you, AJ. I appreciate that. One of the things I was saying earlier with talking to Tony is that one of the challenges of this course, and also one of the virtues of this course, is that we get people who come from all different backgrounds. We have literally over the years had pure mathematicians, surgeons, computational biologists, chemists, MDs who are not surgeons, toxicologists, and we've had everybody from high school students to emeritus professors. And so nobody comes to the course with the same background.
Yeah.
And we've been looking at the polling that we've been doing about modules. And in every module, there's some people who feel it's going much too fast, and some people feel it's going much too slow. Some people feel it's covering too much material. Some people feel it's covering too little. We've thought about trying to fork the class to have an advanced class and a more beginners class.
It's definitely hard to keep up with building the model in real time, and especially when whoever's doing the instruction sort of deviates from what is in the slide or in the uploaded model, which is used as a reference. But in the end, I guess at some point we have to just go back and redo the whole thing, build the model slowly at a pace that works for us. Otherwise, because what I found was I certainly have a little bit of a hard time doing it in real time. And then if I'm engaged in trying to deal with whatever peculiarities of loading, I miss what the person is saying, and then I'm completely lost. So I have to make sure I just keep paying attention, because that's more important than trying to write it in real time, I think.
Right, I think that's tricky. I think coding live is hard.
Yeah.
On the other hand, I think having some exercises is essential. And so
Oh yeah.
balance between those two things.
Yeah.
I thought actually Giulio Belmonte did a very good job.
I absolutely agree. Absolutely agree.
And I think the way Giuliano and Gilberto presented it, if people are familiar with the material already, then that works. But perhaps we're going to go back and rework those slides. So instead of covering that material in one module, it takes 2 or 3 modules, and you have more.
Yeah. No, you're exactly right. For my level of competence or incompetence, whatever you want to call it, Julio's was definitely just the right pace for me. It was very well done. Yeah.
And I appreciate. I mean, we do take those questionnaires seriously.
Yeah.
And so for next year, we'll go back and rework those modules where people seem to have trouble following.
Yeah.
We always try out new ideas. The thing that happened last year was, of course, we taught the whole course around the idea of building a COVID model.
Yeah.
And then this year, people asked us to go back to the more developmental biology focus.
Well, I certainly appreciated that.
But that meant that we had to redo all of the lectures.
Yeah.
So I thought last year we actually had quite a good organization.
Yeah.
The slide decks were very well organized, but we couldn't use them.
Yeah.
No,
and it was a good alternate way of doing it, because you did one very complex model very thoroughly and step by step. But I mean, first of all, it was much more new to me then, so I got lost much more easily. And so this year it's been much better for me.
Well, we appreciate you coming back. I was saying to Tony earlier that I always say the same thing, which is that if something is actually worth hearing once, it's worth hearing twice.
Yeah.
It's either not worth hearing at all, or it's worth hearing more than once.
And this year I have managed to recruit a very nice postdoc who's he didn't actually have that much background in CompuCell 3D, but he's worked through last year's workshops. He's attending again, Abhishek. He's the one who's on my slide. So we'll work together, and we're really happy to have been on this course together.
Well, I appreciate it very much. And again, I appreciate everybody's patience with doing it virtually. As they're doing it in person makes it easier in the sense that if people have issues, in the old days, we'd have a room with 20 people in it. We had a beautiful conference facility for it, with each person at a big table. And so there was enough room that we could, the instructors could circulate and sit with each group.
Right.
And work with them individually. We're trying to do that with the breakout rooms, but it's not quite the same. On the other hand, we're also able to have people from Europe, people from Asia who wouldn't be able to attend if it were in person. So there always are trade-offs.
No, it's true. I think this is the net value of the virtual meeting has been definitely positive. I'd say the number of people who can attend much more easily, not only in terms of the cost, but the logistics, because of COVID has made it so complicated to travel that it would have been impossible.
So there was a question in the chat: are the past schools archived and accessible? The slide decks and the videos should be available. We can they should be on the CompuCell site if you click on past meetings. And at least some of the videos are up on the YouTube channels. The main problem is that we don't have funding for supporting the sort of the AV and things. It's quite time consuming and expensive to edit the videos. And so we need to go back and do some more editing of the videos. But yes, the past workshops, last year in particular, all of those the videos are available, and all of the slide decks are available. So if you're interested in all the exercises. And so in last year, we started out with a viral dynamics model, an OD viral dynamics model of a simple infection, SIR type in-host model, the kind of a model that people like Alan Perlson write. And then we took that model and converted it step by step from being an OD model to being a spatial model of a patch of tissue being infected. And
that's great. I have another question, and this is to do with the parameters. When I deal with multi-parameter system, the right, in quotes or not, parameter set is not unique.
That's absolutely true.
And the only way to verify that it has certain reality to it is by comparing to experiment or to the literature in large. And that brings a lot of questions, because some of the goals of these simulations is to save time of expensive experiments or to design the next ones.
So those are very fundamental questions, and I'd love to. I have a whole series of lectures on those issues, which I'm not able to give because there's no time. If we were when we do this in person, we actually have more. The days are quite a bit longer, but we figured that 6 hours of sitting watching Zoom a day for 5 days plus 2 plus 1 for eight days was enough. And so
more than enough.
Right. When we do it in person, we typically start at 9 a.m. and end at 6 p.m. And then we have a little bit more time to cover some of these more very important issues. These models are always overfitted. There are many sets of parameters that will do the same thing. And there are some fundamental questions about how you do validation. One of the reasons we did the course last year around doing an ODE model first and then turning it into a spatial model is that fitting the parameters for the ODE model is easier. And then you can actually know that the parameters, at least those parameters, are correct. And so there are a whole series of issues about how you do parameterization of these complex models. If you're interested in hypotheses about whether mechanisms exist or not in a system, then often the exact parameter values matter less. But it's absolutely true. I'm delighted to have some questions because the whole point of this early period was to have this kind of conversation. So I'm glad that on day 4 we're having it. This kind of issue is profound, and I don't have a good answer for you. Something that we've been doing a lot more of is that we typically, the CompuCell was designed so it would run on your desktop in real time so you could see your results and play with them. And so it was really created to be an exploratory tool. However, these days we tend to run it on large clusters. We'll run tens of thousands of replicas, hundreds of thousands of replicas. And so, for example, when we showed the COVID simulation where we did a remdesivir dosing simulation, that would involve thousands and thousands of replica simulations where each parameter set was run multiple times because they're stochastic models. And then you would then scan multiple parameters simultaneously. Josh and a team at Pittsburgh have been building a nice interferon model. And again, what they did was they started with a experimentally validated ODE model of interferon response, and then asked the question, when you move that non-spatial model, which is deterministic and doesn't take into account heterogeneity in tissue, and you put it in a spatial context, what changes? And so they were simulating plaque assay, viral plaque assay growth in the presence or absence of interferon pre-stimulation. And in that case, you can nail down at least some of the parameters pretty well. So it very much depends on what kind of experiment you're talking about, what information is available. Another slogan that Tony's heard and AJ's heard is that building these kinds of models is brutally revealing of what you don't know biologically. And very often when people come to my office, I used to have a wet lab. Since COVID, my wet lab is gone. But I did plenty of developmental biology experiments in my day, and I did microfluidics, these kinds of things. But you would say to people, they'd have an idea about a project, and you'd say, these are the things you need to know to be able to build a model. And the response would be, well, if I knew those things, I wouldn't need the model.
Yeah. Another question, if I may.
Please.
Can I take distance and think about doing PCA analysis in the parameter space?
You certainly can. The only problem with that is that the simulations tend to be long, and so you have to think about it efficiently. I mean, one of the big problems with using these kinds of models is if the simulation takes something. There was a question in the chat about can you multi-thread CompuCell? The answer is yes. If you have a high-performance computer with 64 cores, you could say run it on 128 threads, and it'll run about 60 times faster. And that's an option in the beginning of the XML, the CC3D ML, the markup language. You could switch, and that works really well on high-performance computers. But I was going to say, one of the issues that we very early version of CompuCell actually had an MPI deployment where you could actually spread out one computer simulation over many boxes, and people didn't use it at the time. This is 20 years ago now. But when you're doing optimization, you have to do sequential runs. And so if each simulation takes 2 hours to do, and you have to do 1,000 generations of optimization because you're doing something with AI, or you're doing something with even a classical optimizer, then that means that optimization takes 2,000 hours to run. And so we tend to use things like particle swarm methods, which are less efficient in terms of total computation time, but are more efficient because they require fewer generations to converge. And so you have to think about how to do your analysis. There's a paper that Jim Sluka did with Xiao Fu, who's now at the Crick in London, who's a graduate student, excellent graduate student in this group, where they did do detailed sensitivity analysis of their liver toxicity model. But that's single factor analysis. That's not doing full PCA. And the interferon paper that Josh has done, they also did sensitivity analysis, but again, not full PCA. But there's certainly no reason. CompuCell now is callable. It runs on a cluster. And so provided that you can return a metric from your simulation that gives you a residual, you can call a CompuCell simulation from a Python library that does PCA and returns a residual. It'll take a long time to run, and you'd like to have one that uses a cluster deployment so it can run multiple replicas simultaneously, but that's possible. That's outside of the scope of this lectures this week. Although Jim will talk maybe a little bit about cluster deployments. TJ could do that. But if you're interested in how to do that, TJ has thought about it a lot. And we'd certainly be very interested in seeing more formal ways of doing it. So I'd be very interested to talk to you about it. Any other comments, questions?
Thanks for that long answer.
I'm sorry, I didn't mean to go on too long.
You're doing very well. I'm not in position to give you compliments, but you're doing very well. By the way, I was lured to the whole thing by your talk, I think 2 weeks ago in Toronto.
Oh, well, thank you.
And Maria, I'm not sure if Maria has the time to participate. I didn't see her or heard her, but I'll report to her about the experience, which I have in the so-called no hands-on mode. I'm the one who is telling you all the time, not doing the exercise, because as AJ says, I'm trying to think what's going on here in the big picture. Okay, the details I may or may not ever even deal with, but that's a different story. So Maria, I'm sure will appreciate this information, and I'll make sure that she's still with it.
I know she showed up the first day. I saw her on the call the first day.
Yeah,
I don't know if she had it.
That's called time pressure of managers.
Yes, well, that's the one advantage. That's another advantage of doing it in person, that you're not distracted as much.
Sure. Yeah.
It is hard to do it, and I appreciate people's patience because I wind up running the Zoom sessions, which means I don't have as much time to interact with people because I'm essentially having to manage the meeting, and it does take some time. Pedro's been doing a great job on the meeting management, and Giuliano as well. But it actually does take some effort to manage the Zoom, and that's we appreciate that. So there's another question. How do you fit the value of the energy for different cell types? So traditionally, we would start off this course with what a physicist would call the hydrogen atom, which is only a joke a physicist would understand. which was the cell sorting simulation. And I actually showed that at the very beginning of the first lecture. And we moved away from that for all variety of reasons, mainly because it was a bit of a dead end in terms of where it went. But the cell sorting simulation asked the question, and it's what the whole CompuCell thing started back in 1993, actually earlier than that, 1992 even, when there was a series of experiments. There's a classic series of experiments going back more than 100 years, where you take two cell populations, embryonic cell populations, you mix them together, and they sort out so that one forms a clump in the middle, and the other goes into a disc around the central clump. And the late Malcolm Steinberg from Princeton, who was a wonderful man that I worked with, I never worked for him formally, but he was sort of a mentor to me for many years, did a whole series of experiments that showed that the sorting order of the tissues corresponded roughly to the adhesivity of the tissues, the contact energies. And he came up with what was called the differential adhesion hypothesis of embryonic organization. Now, in reality, embryonic organization is more complicated than that. In the first place, when two cells come together, the adhesion between them changes over time as they form junctional adhesion structures, as the cytoskeleton changes, and so on. And secondly, cells aren't passive. And so there also are things like apical rings, contractile rings, and there's active contraction. So there's also a differential contraction hypothesis. And that's especially prominent in Drosophila embryos, but happens in all embryos as well. But the original version of CompuCell was written to simulate differential adhesion, reorganization of tissues under differential adhesion. And what you find is that what matters is the order of the energies. The most cohesive things go to the middle. The least cohesive things go to the surface. If the adhesion isn't sufficient, the cells spread out the way Giulio showed in his demo on the first day. And so you have a situation in which the order of the energies matters a lot. And if you make the energies too large, you can have artifacts where things freeze on this lattice that we work in. If you make the energies too small, you have situations where the cells fall apart into dust. But over a very broad range of energies, surface energies, contact energies, provided that the order of the energies is right, the more cohesive things have lower contact energies, the less cohesive things have higher contact energies. As long as that order of energies is right, you get the right dynamics. And then you can fine tune it by doing parameter fitting to get the exact ratios. And so that's actually a nice set of exercises to do, but it's time consuming because the cell sorting simulation, see, these days with modern computers, it runs quite fast. In the old days, running one of the simulations took quite a while, and so running enough of them to explore that parameter space was time consuming. So does that help a little bit with that? But one thing that we've talked about for years and never quite gotten to is to switch from this contact energy plug-in to this contact local flex, which is based on the number of molecules of each type. And that's much more natural because then each cell can have a different number of cadherins or a different number of integrins. And there's nothing intrinsically difficult about it, but you have to calculate then, if I want this energy hierarchy and the functional form of the adhesion is this, then I have to back out what are the number of molecules I need, what are the parameter values. And that it's not hard, but it's a calculation that takes a while to do. And we could either show you a result and have you do it, in which case you don't learn very much, or we could walk you through how to do the numerical calculation but that would take an hour and a half to just show you how to do that calculation. It's not hard, but it's arithmetic. And so for the purpose of this course, we decided to stick with the older system of contact energies. But for example, I have a slide deck on cancer evolution, simple models of cancer somatic evolution, where the adhesion between the cells is determined by the number of cadherins and integrins. And then as the cells mutate, they change their numbers of cadherins and integrins, so the amount to which they adhere or don't adhere changes. And so that's actually a pretty simple example of using that. And the manual has a pretty good demo of that, of how to do it. And so that's something, if somebody's doing a project, a hackathon project, we could definitely work with people individually on how to do it. We need to make some slides and a tutorial on how to do that, probably. It's a good suggestion. There was a question about increasing the output frequency. Will your simulation be faster? No. Oh, well, okay, there are two questions there. One problem in CompuCell is that we're a little bit trapped by some of the terminology that was written down 30 years ago when we didn't know this would be continuing. And so there are terms like temperature that really shouldn't be called temperature, but we're stuck because that's what everybody in the field uses now. Another thing that happened in CompuCell is that there are things called frequency, which are actually periods. So the output frequency is actually the interval, the period between outputs. And so the question is, if I increase the output frequency, will the simulation run faster? If you actually increase the frequency, the simulation will run slower because it takes time to draw each image. If you increase the number of the output frequency, which is actually making the frequency lower, increasing the interval between outputs, it will speed up the simulation. And one thing that's a minor annoyance of Python is that the print statement takes a lot of time to execute. I don't know how that happened. It doesn't take time in C++, it doesn't take time in Fortran, but it does take time in Python. And so when you're printing a lot of material to the console, that takes some time, and you're usually better off debugging with that, but then turning those print lines off if you're going to run in production mode. All right, any other questions? I'm really glad that we're having such a lively discussion this morning. I appreciate it very much.
I had posted a question earlier on, but maybe you can address it later. It's about comparing CC3D and Morpheus to understand the strengths and weaknesses of both. So it can help us decide for a particular problem whether one platform is better suited versus the other.
So we work a lot with the Dresden people. They're good friends. And Morpheus has a beautiful scripting language. It has its own scripting language, though. And the advantage of the Morpheus language is that it's very elegant, and it's in many ways very simple. And there are certain functionalities that Morpheus provides that are simpler to use than in CompuCell. The disadvantage is that because it's a unique scripting language, it doesn't integrate as well with the functionalities that Python has. You don't have an import NumPy. And so I would say that we've actually tried to learn from some of the things Morpheus has done. And Giuliano tried to write a converter between the Morpheus model specification and CompuCell. And you can actually convert quite a bit, but not all of it.
Yeah, the hardest part are events, so if statements in Python. There are ways to do that in Morpheus, but I don't really remember how to. But that conversion is tricky and hard to do.
But just before the lockdown happened, we were actually in Dresden. for a week working on this problem. And
then-
I would say that CompuCell is a little bit more flexible,
Mhm.
and it does support some features that Morpheus doesn't have. So
Yep.
so for building models quickly, they're probably comparable. Maybe Morpheus has some advantages.
Yeah, I think Morpheus is a bit easier.
But I think you bump into some limitations. Although the Dresden people are great, so if you point the limitations out to them, they'll work and extend the
Yeah.
code. But we've dreamed for a long time of trying to merge the two packages, because there are these unique features on both sides that are really very attractive.
So I had a particular question, in terms of some of the things we're trying to do where we want to get apical constriction.
Yes.
With that particular phenomena, how do they, because in CompuCell you suggested that we need to make a hybrid cell of lateral cells and apical cell and a basal cell.
Yes.
Is that different in Morpheus, and is that a different type of solution to that kind of a phenomena?
I don't know. I apologize. I don't know how they do it. We will show how to do it tomorrow.
Yeah, I'm looking forward to that.
Okay. All right. Well, I guess actually it's 10:30, so it's actually our official start time. Let me quickly walk through our outline for today before we get into the main lecture. Appreciate everybody coming back. Very much appreciate you sticking this out. Our schedule has changed a little bit, clearly. We are going to start now with module 3.4. And even though it's in the place of module 4.1, it's going to keep the number 3.4. And so I strongly encourage you now to download the demo. Juliana, if you would post the link to the demo again. Download the demo, and if you're using NanoHub, please upload it. I will quickly walk you through that, but this whole lecture will be built around that demo, and it's critical to have that demo up on your computer if possible. Okay. After that, we will have a hackathon teaming discussion. Somebody asked if they could do updates on the hackathon team results. And so we're going to do that. We're going to have first 4 additional hackathon ideas from people who wanted to present. I hope they're going to be here. They said they would. Who wanted to present Tuesday and weren't able to. And then I will run through the slide decks for the people who presented before. And if you want to ask questions, make comments, suggestions, that's fine. If you want to set up some breakout rooms to meet with people individually, that's fine too. Anything we can do to help with teaming, that'll be good. And then whenever we're done with the hackathon section, we will go forward and begin to build subcellular network models using the Antimony tellurium language and coupling them to cell behaviors. And so Joshua will be showing those. Depending on how that goes, we will either show how to do Boolean network modeling integration using MaBoSS, and that may either happen this afternoon or tomorrow morning, depending on how the schedule works out. So again, I will start my lecture in just a minute, and then we will have our usual break. We'll have the hackathon teaming. When we're done with the hackathon teaming, Josh will take over and begin his presentations of the subcellular model integration. All right. Are there any other questions before we start? Again, this module is still numbered 3.4, even though it's in the 4.1 slot. So the schedule will be slightly out of order. We're going to be module 3.4, module 4.2, and then continuing with module 4.1 because we ran over a little bit yesterday. Each year we try out some new lectures. Some are wonderful, some need improvement, and that's why we keep asking you these questionnaires about your experiences. All right. So now I would like to move to I'd like to move to a discussion of networks. Networks are big. Is that screen share working?
Looks fine.
Yep.
Okay.
Good.
Thank you very much. Okay. Well, welcome back, everybody. This is module 3.4 on the CompuCell workshop. It's going to be an introduction to network modeling inside of CompuCell 3D. I remind you that the lectures are being live streamed and recorded, so please be aware of that. Network models are a very large topic, and in fact, Herbert Sauro 2 weeks ago spent an entire week teaching the subject. I know some people in the class have taken that course. Others have maybe learned this material in other ways. I'm going to talk very briefly about a few issues that you might want to be aware of when you're doing network models. And then we're going to focus on how to build network models inside of CompuCell. And so I'm going to cover this in a bit of a hurry. And there are going to be a lot of issues that are of interest and importance that we're not going to be able to go into in detail now. I'm also going to say in advance that the particular simulation that we're going to use in this module has some concepts and materials in it that we're not going to cover right now and that Josh will cover later in the day. So I'd ask you to focus on the network part of the simulation and not worry right at the moment too much about how the back-end magic of the simulation works. So we're talking, this course was always called Multiscale Modeling, and we've seen slides like this before. The body has many, many levels of organization, from individual molecular reactions, for example, in metabolism, through the macromolecular machinery and transport that goes on inside of a cell, through the organization of cells into tissues, tissues into organs, macroscopic transport, for example, in the blood and the lymph, or in air, in the lungs, and then large transport by blood, for example, between organ systems. And so we have always many, many scales to think about. Typically in CompuCell, we're working at the level from subcells to cells to tissues. We're neglecting the complexity of macromolecular machinery and a lot of the structure within cells. Although yesterday we saw a simple model where the cell was broken up into 3 compartments, a nuclear compartment, a lamellipodium compartment, and a cytoplasmic compartment. And one can go a lot further in that kind of development if one wants to. I'd like to say again that this is going to be a very quick overview of some very important concepts. I strongly recommend the manuals that Herbert has written for tellurium antimony. The Tellurium Read the Docs has a great tutorial with online executable code for doing simple network simulations. I've mentioned before Herbert Sauro's textbook, Systems Biology and Introduction to Pathway Modeling. That's a very elementary book, but covers step-by-step in real detail how to start building these kinds of models. And we have in our Google Drive the Tellurium Antimony cheat sheet that gives you the basics. And again, I encourage people to print that out and have it on your desk to remind yourself of the basics. Although I have to say the antimony language in some ways is so simple. The reason we use it is because of that simplicity. it really makes it easy to build this kind of model. Knowing what model to build is not necessarily so easy, but implementing it is. Farouche asked, could you please send the link to the live stream? Okay, let me do that. Does somebody else have it? Okay. There's the live stream link. Okay. And if you want to learn more about the topics that we're only going to be touching on today, there are many, many textbooks and places that you can go. These topics would include things like dynamical systems, the theory of stability of networks and fixed points of networks, bifurcation theory. There are books on how to calculate networks and infer networks from experimental data. And there are wonderful books on mathematical biology of all kinds. And again, I could make some suggestions offline if Herbert's textbook isn't the one you want. Herbert's book has some very good references in it as well. We're just going to be talking about some very simple ideas today, and I'm going to skate over a lot of the complexity because the goal here is to get you writing these models. If you've done this kind of modeling before, I hope you'll find it relatively easy to do it. If you haven't done it before, I hope the examples are simple enough that we can work through them together. So when we think about biological networks, there are four main kinds of biological networks that we're going to deal with. There are chemical networks and metabolic networks where we are creating and destroying molecular species. And the classic example of those would be metabolism in the mitochondria. Those tend to operate on very fast timescales. There are signaling networks that transfer information within and between cells, things that would involve molecular receptors on the cell surface, G proteins, and so on, kinases. Those tend to operate on a slightly slower timescales than the metabolic networks. They're regulatory networks that turn gene expression on and off or do post-transcriptional modification. And those operate typically on slower timescales still, timescales of 15 to 20 minutes. So metabolic networks, we're typically talking about tenths of a second or less. Signaling networks, we're talking about seconds to minutes. Regulatory networks typically operate on the timescale of 15 minutes and above. And then there is a whole separate question of building models of macroscopic transport, which are used in things like drug discovery. And those are physiologically based pharmacokinetic models. And those typically have a mixture of timescales. Blood transport is very fast, but things like elimination of chemical species are quite slow. And they have their own world. The mathematics is the same, but they have their own world about how to do parameter fitting and so on. But all of these models, all of these networks can in fact be implemented computationally in the same way and mathematically in the same way. These networks tend to operate together. Excuse me if I'm drinking about a little hoarse this morning. And so we want to ask a lot of questions about how networks operate. We can ask the question, does the network that's active inside of a cell change? When a gene is turned on or off, you have molecular species that can interact that weren't there before. When you have a cell that has a particular set of networks active, the state of the network could change. You could have more of a phosphorylated version of a protein or less. And of course, the components of the cell themselves could change because you could build additional mitochondria. You could change the structure of the cell. And typically, we have to think about all of these networks together. However, very often in the simulations that we're doing, we're going to assume, just the way when you drive a car, as long as the car is working properly, you don't worry too much about how the engine works. For many of the things that we're going to do, as long as the cell is alive and doing whatever it does normally, we're not going to worry so much about metabolism. If we're interested in the production of toxins by as a metabolic byproduct, then we'll model that. But the basic metabolism of the cell is something we typically have hidden under the hood, so to speak. So if we talk about chemical reactions, it's interesting. I'm sorry, PowerPoint has hidden a slide hidden a figure from me. I apologize. We are talking about transformations, chemical transformations. If we think about a chemical reaction formula, here 2 ADP go to ATP plus AMP. That's a reversible chemical reaction, or some kind of system of chemical equations. 2 molecules of A go into B. B can make 3 of C. C and A can combine to make D, and these are reversible. What we'll see is that we're used to writing chemical reactions in the form of arrow diagrams. These chemical reaction formula are in the form of a substrate on the left and a product on the right. We get rid of 2 ADP and we get 1 ATP and 1 AMP. On the other hand, we could run this backwards and take an ATP and an AMP and turn it into 2 ADP. We're not talking about the energy. So then when we think about a network more generally, we have to think about the nodes. Those are the values that sit, A, the amount or concentration of chemical A, the amount or concentration of chemical B, the amount or concentration of chemical C, and arrows. And in this case, these arrows represent chemical transformations. We destroy A and create B. We destroy B and we create C. We combine A and C, which destroys them, and create D. Now, of course, chemical reaction conserves mass, but the key thing to think about in a diagram of this kind is that we have A goes to B, and it has a rate, and these are called rate laws, and the rate tells you how fast A is used up and B is created. Classically, in simple chemical reactions, the simplest form of chemical reaction rate law that we have is called the law of mass action, which says that the rate at which we take A and B and produce C is a constant times the amount of A times the amount of B. That is, it's bilinear in A and B. At least for the case of not having a lot of A and B, that makes sense. If we don't have any A, we can't produce C. If we don't have any B, we can't produce C. And if we think mathematically in terms of a Taylor expansion, the lowest order term is going to look like that. Now, when we have very high concentrations of A and B, typically we'll get some kind of saturation, but at least at low concentrations, mass action works pretty well. And there are whole textbooks which assume everything in the world is mass action or a simple generalization of that. It makes the mathematics a little bit easier. But one thing to remember is that this one arrow diagram, A plus B goes to C at a rate VF of AB, actually generates 3 differential equations. One for the amount of the concentration of A, one for the concentration of B, and one for the concentration of C. And if you're used to writing ODEs for rate laws, you have to remember that when you start writing rate laws in terms of arrows, the number of equations that you write is slightly different and their form is slightly different. And we're going to be writing things in terms of these arrow diagrams in antimony. The natural language of antimony is arrow diagrams. Now, in reality, life is more complicated. Metabolic networks are usually segregated in space. And within the cell, there are all sorts of complicated structures like the endoplasmic reticulum and the Golgi apparatus, which subdivide the cell into compartments. We're typically going to assume that, as most network models do, that the cell behaves like a stirred reactor, and we're going to neglect that spatial compartmentalization unless we're going to model something very specific. And so we're not going to think about spatial structure to begin with. Although in the example we showed yesterday, we could perfectly well have different chemical reactions happening in the nuclear compartment, the cytoplasmic compartment, and the lamellipodium compartment. The next kind of network that we have to deal with are signaling networks. And a classic example of this would be Notch. Notch is a transmembrane receptor. When it binds on another cell to delta, it's cleaved, and the intracellular component of Notch then goes into the cytoplasm of the cell and sends a signal that, among other things, can control cell proliferation. Another example of this would be the classic phosphorylation kinase cascade of MAP kinase. We start out with MAP kinase, which phosphorylates MAP kinase, which phosphorylates MAP kinase, which phosphorylates a bunch of other stuff and does things. And so that kind of kinase ladder is something that we'll model a lot. Signaling networks are things that we're going to want to be able to model. And one of the things that we have to think about in a signaling network diagram is that the signaling networks don't have the same transformational structure. The arrows mean something different. And I'm going to talk a little bit about arrows just to help people out, because there was a request for that from somebody before we started the course. The kinds of questions you might want to ask about a protein signaling network would be something like, if I increase the amount of vascular endothelial growth factor, VEGF-A, in the environment of an endothelial cell, vascular endothelial cell, does the cell proliferate or does it die? Depending on whether it's a physiologically meaningful concentration, you could get both responses. What happens when the number of receptors on the cell membrane changes? How does that affect the cell's migration? If I mutate a component of this network, what happens? And if I mutate that component, does that lead to a change in the cell behavior that leads to a disease state? That's very important if I'm thinking about cancer. A note about networks, protein signaling networks. Typically, you'll see these lollipop diagrams, lollipop arrows for activation, these T-shaped arrows for inhibition. We'll use the pointed end arrow for mass transformation. And links come here. Typically, we're talking both about chemical transformation. When a molecule is phosphorylated, that's a chemical transformation. But the kinase that's doing it may or may not be molecularly, well, if it's a kinase, it actually is reacting because it's transferring the phosphate. But in many cases, we'll have a situation where we'll see a diagram like the one on the top right, where we have S1 is transformed to S2. S1 could be the non-phosphorylated form. S2 could be the phosphorylated form of the molecule. And A here is promoting that phosphorylation. In this case, it's a regulatory arrow. A is not being used up in this reaction. Similarly, we could have R, which is inhibiting this transformation. And regulatory links don't use up typically the components, and we have to think about how to represent that. And what we have to think about then is if we have S1 being turned into S2, and that's promoted by A, what that means is that the rate of production of S2 increases as the concentration of A increases. Sometimes this diagram, A promotes the transformation of S1 to S2, is written with a shorthand that just says A promotes S2. You'll see that a lot in biology textbooks. When we see that, we have to interpret it in the following way. We don't know what was being used up to produce S2. And so we have to think about this diagram, A promotes S2, as nothing, some unknown thing, produces S2 at a rate regulated by A. As a chemical reaction, that means nothing goes to S2 at a rate that depends on A. Similarly, if R inhibits S1 going to S2, that's sometimes written with shorthand, R inhibits S2, that's interpreted as a chemical reaction, nothing goes to S2 at a rate regulated by R. And as a chemical reaction diagram, that is, nothing goes to S2 at a rate V1, which is a function of R. And so we'll see a lot of what are called dangling arrows, where we either have nothing going to a chemical species or a chemical species going to nothing. It's not very different when we talk about gene regulation, which describes how genes are turned on and off. There are these standards for how to diagram gene regulatory networks. They make things look very simple. In fact, they're not as simple as these diagrams suggest. Gene regulatory networks typically control long-term changes of cell behavior, especially things like cell differentiation, although they're not the only thing that happens in cell differentiation. And so we could ask the question about, if we change the amount of a particular protein, how does that change the amount of messenger RNA being produced? And then downstream, how does that change the amount of the protein coded by that messenger RNA? What happens when we knock out a gene or knock in a gene or make a particular mutation to a gene? Does a network have a stable state which we could describe as a cell type? Does a network allow for differentiation? And of course, very often we're interested in whether drugs change the gene expression in these kinds of networks. Sometimes we're also interested in toxicity. A lot of toxicity studies look at the effect of particular molecular species on gene regulatory. The typical diagram for a gene regulatory network looks like this. Here we have a molecule I, it's an inducer. It acts on gene P. Gene P produces messenger RNA, and the messenger RNA is then translated to protein. Notice that I is not used up in this. The gene is not used up in this. The messenger RNA is not used up in this. And the protein is not a product directly of the messenger RNA. It's produced by the ribosome. And there are a bunch of steps involved. The inducer has to bind to the gene. The gene has to be transcribed to messenger RNA. Well, transcribed to RNA. The RNA has to be edited to messenger RNA. The messenger RNA has to be transported to the ribosome. The ribosome has to translate the messenger RNA to the raw protein. The protein has to fold, and then there's post-translational modification. So there are a whole series of steps that are hidden in a diagram of this kind. Very often that mRNA is hidden as well, and we might even see just I promotes P without going through those steps. If we want to think about translating that, we'll see in a book, P1 promotes P2. P1 is some kind of transcription factor. P2 is a protein. What's really happening is P1 binds to a regulatory site on the gene. The gene is transcribed. This would be is transcribed and then translated to produce P2. As a chemical reaction, the things that are used up are not modeled. So we have nothing goes to P2 at a rate that depends on the amount of P1. Very often we want to think about the fact that the protein will decay. And so we'll also imagine usually a first order decay kinetics where the protein turns into nothing again. So this simple diagram, P1 promotes P2, actually turns into nothing, turns into P2, and P2 goes back to nothing. And what's important are how these rates depend on the amount of P1 and the amount of P2. If we wanted to include the messenger RNA step, because that has a significant delay, then we would have the promoter binds to the transcription factor promoter binds to the activation site. We have transcription to messenger RNA. The messenger RNA itself can decay. The messenger RNA can be translated to P2, and P2 can decay. So this diagram, P1 promotes P2, actually then translates into 4 reactions with 4 rate laws. And so that's something that we always have to remember when we're going from the biological description to the mathematics. Typically, when we are going to interpret P1 promotes P2, we don't know the details of how P1 turns P2 on. Sometimes we'll use Boolean models of that. In continuum ODE models, typically we will assume a Hill function, a sigmoidal rate of production of P2 as a function of the amount of P1, with a basal rate of production, B1. And the steepness of this curve is controlled by this exponent n, and the half maximum concentration for P1, at which we produce at half our maximal rate, is defined by a Hill constant K1. We'll use typically the balanced form where we exponentiate K1 with the same exponent as P1. The steepness of this curve is determined by the cooperativity of the reaction. That could typically be 4 or maybe 8 for steep reactions, more Boolean networks. One way, if you want to build these in a consistent way, is to always rescale reactions between 0 and 1. In that case, K1s are always set to a half. And you can have increasing functions for activation. If P1 inhibits P2, the nice thing about Hill functions is that all you do is you change the numerator from being P1 to K1, and now you get a decreasing sigmoid with the same half maximal value, and the functional form looks the same. And so if I were going to turn P1 into P2, then if P1 inhibits P2, you will get an ODE that looks like the rate of production of P2 is some basal rate of production with a decreasing Hill function, and then P2 decays. Gene regulatory networks come in motifs. There are a lot of classical motifs, Herbert's textbook, and there are many textbooks that talk about some of these. The simplest ones are gene activation or repression of a single gene, but you can have 2 activators or inhibitors that act together. You have to then think about how they combine. You can have a gene cascade where transcription factor 1 turns on gene 1, which produces another transcription factor which turns off gene 2. You can have autoregulation where a gene produces a transcription factor that turns itself off. The effect of those transcription factors can be modulated by other molecules, and they're very, very often regulated by phosphorylation. Typically, because of the time it takes to do transcription and translation, that's typically 20 to 40 minutes for a typical protein. These networks operate on a fairly slow time scale. If we now jump to the blood transport in the whole body or lymph transport in the whole body, these are now physiologically based pharmacokinetic models. These are also network models. In this case, we're modeling flow. Typically, what we'll do to begin with is draw compartments, and these would represent main organ systems: the brain, the kidney, the heart, muscle, fat, other tissue. Here's the liver broken out, the gut, the lung, and the blood as these 2 compartments, the venous blood and the arterial blood as separate compartments. In this case, the basic model structure is a transport equation that says how much material the blood is carrying at any given time between the heart and the venous blood, or between the venous blood and the lung, and so on. And we might also want to know how molecules are being absorbed, eliminated, or absorbed by these various components. And these typically would be asking questions like, if I take a pill at a time X, how long does the pill take to get into my bloodstream? How long does it take to be eliminated from my bloodstream? What fraction of the molecule in the pill goes to particular organs and particular targets. Numerically, these are fairly simple models. We have these fluxes Q that describe the transport between the various components. In this case, there's an explicit picture where the kidneys are excreting the molecular species into the urine and eliminating it. Here, the gut lumen, your intestinal contents, are being absorbed by the gut. The liver is breaking down the molecular species, putting them out in the bile for excretion in the feces. This kind of structure is also a network. Molecularly, these are quite simple. You have typically what are called Fickian laws, where you have transport, forward and backward transport, that's proportional to the concentrations. And in the case of these kinds of models, you typically actually convert concentrations to amounts. You have to know how much the volume of the blood is, the volume of your various tissue and organs are to do that conversion. But again, you can write everything in terms of an arrow diagram. Blood, molecular species in blood is being transformed into a molecular species in body. Molecular species in body is being transformed to molecular species in blood. So all of these kinds of networks can be written in terms of arrow diagrams. Of course, in reality, all of these things work together. The cells wouldn't be alive if they weren't doing metabolism. They're continually receiving signals and sending signals to the outside world and signaling within the cell. Their gene activity is continually changing. And of course, to supply the metabolism of the cell and to get rid of the cell's waste products, we need transport through these PBPK networks. And so that's lightning fly-through of what would probably take a week or two to go through in a regular course. But I'm assuming that people have had some familiarity with this kind of work. On the other hand, you may, if you're coming from a biological background, not have thought so much about how to translate those arrows, which are the classic language biologists use to represent these models, how to translate those arrows into mathematics and then into code. So I did want to show you a couple of rate equations so that you have an idea of what they look like. Okay, so now let me see if this will work for me. I'm going to now split screen. Can people see both screens okay? No.
No.
Okay, let me reshare. Thank you. How about now? Okay, let me do a quick poll. I meant to do this poll earlier, but I guess it's a little bit late, which was to ask people how much network modeling experience they had. I meant to do that at the very beginning of the class. While we're doing the poll, please download the example from the 3.4 folder. If anybody is having any trouble finding that example, please reach out to Giuliano or Jim Sluka and get some help, because if you can't do it, it's going to be boring for the next half hour. And then if you're using NanoHub, please upload it to NanoHub. I will walk you through that, but it would be helpful. Okay, so if I look at this, the responses to my questionnaire, a few people haven't answered yet. It looks like about half of the people have done some kind of network modeling, and about half of the people haven't. And so my guess is that if I do a poll on the module, the people who said that they'd done network modeling before will say I went too slowly, and the people who haven't done it will say I went too fast. For both sides, I hope for the people who have done it before, there was some material in there that was presented differently. And for the people who haven't done it before, I ask you to hang in there. As I said earlier in this class, think if something's worth hearing once, it's worth hearing twice. And so you'll hear these things again. And so nothing else, I hope you got pulled away from the presentation. A few key concepts of different kinds of networks and the idea of a rate equation and the idea of using chemical transformation diagrams to represent different kinds of biological network. Okay. Oops. Okay. I don't know why Zoom decided to change my my screen there. Okay, so now what we're going to do is I'm going to do a very, very simple introduction to writing these kinds of models inside of CompuCell. And then in the afternoon or later today, Josh will work in much more detail about how to do this. We will use a specification called Antimony to define these models. And we will then convert those models into something that's executable. In this lecture, we are not going to worry about how that loading is done and how that conversion is done. That will be left to the afternoon. Again, on tellurium and antimony, please go to the tellurium, read the docs, and please do print out those handouts, those cheat sheets that we sent you earlier. Okay, so the first thing that I need people to do if you're using NanoHub, if you're not using NanoHub, you don't have to do this. It's easy. Once you've downloaded the file, you're done. But if you are using NanoHub, please go to the demo folder, download that demo, and then use the Jupyter package or the tellurium package, whichever one you like, to do the upload. So launch Jupyter Notebooks, and then once you've launched Jupyter Notebooks, hit upload. Once you've hit the upload, find the zipped file that is the demonstration. That's called tellurium. And then it will ask you to do confirm the upload. And then once you have done the upload, please switch inside of NanoHub to CompuCell 3D v4 main tool. If you're using this on your desktop, simply open in Twidit the file tellurium.zip. And I'm going to be checking because it's critical before I go on that everybody have that. Okay, I'll wait just another minute or two for people to catch up. If you're having problems, please reach out in the chat. The demo is called tellurium.zip. It is in the folder module 3.4 exercise. Is anybody else having trouble finding it? And yes, you have to upload it exactly the way we've done it before. Looks like 26 people have got it. Let me do it again and just make sure that everybody's okay on this. Can I ask the people who are needing more time to please reach out to one of the instructors for help? Please join a breakout session and get some help. If you're running on desktop, all you have to do is download that file and open it and twit it, so you can skip the next couple of steps. Somebody asked, where is this file tellurium.cc3d? Please reshare. Giuliano, could you please reshare the link to that file? Tarim, thank you very much. Somebody asked for help. Polly, thank you very much for posting the links. Appreciate that. Could somebody join a breakout room? Jim, could you possibly join a breakout room? Is it Igniola needs some help? Would you be willing to take them to a breakout room? You should have access.
Sure. What was the name again?
I will.
Okay, I see it. I see it. Okay.
Does anybody else need help? I don't want to leave people behind, but I also we've got a lot to cover. And so I want to make sure that, but if you can't get this to run, it's going to be boring. Okay, looks like I can move on. Okay, so now I want you to go to CompuCell, switch back to CompuCell. So please switch back to CompuCell. NanoHub seems to have clobbered my CompuCell. So let me go back to CompuCell here. Okay, is anybody having trouble? I seem to have a little bit of an issue with the screen. Does anybody having trouble launching CompuCell? So come back to CompuCell. Inside of CompuCell, select Open Project. Here I'm in Twit. So actually, if you're in, you can do it from either Twit or CompuCell, but launch Twit is probably easier. And you should go to your directory, and there will be a folder called tellurium.zip. When you open that, it will ask you to save it, to unzip it. It may give you a warning that it doesn't have a unique name. Once you've unzipped it, you have to go back to the open and select open again. I've already unzipped it, so if I unzip it again, it'll throw an error. So I'm not going to do the unzip myself. But again, here you'll get this window. Select tellurium.zip, hit open. Which I'm not going to do because I've already unzipped it. If it gives you an error, throws a warning, it should still unzip it. Go back, select the unzipped module, which is now called Tellurium. And it may be hiding one level down, and select CC3D, tellurium.cc3d, and you should be able to open it. So I'll wait until everybody has that running. If you're doing this on your desktop, open tellurium.zip in Twit it, unzip it, and launch it. So let me make sure everybody has it launched. Okay, everybody's got it. Okay, good. I know it's not the most convenient thing in the world, but people people will get used to it. It's not so bad once you've done it once or twice. Okay, so everybody says they've got it. Then once you have it, click on, actually I have multiple copies of it, don't I? The one that I have here is this one called Simple Infection Model. Okay. So you can, I'm sorry, let me find the one. Sorry, I shouldn't have had multiple ones open. This is the one, no, which is the one. I have to find the one Josh wrote. Oh, here it is, the bottom. Here it is. This is what you should have. Let me do a little screen rearranging here so we can see better what we've got. Okay, and double click on Tellurium CC3D, select Tellurium Steppables. Okay, make sure everybody's got that. And go to the top of Tellurium Steppables and you should see this. I'm gonna maybe change the size of my window a little bit here so I can see better. So I want one more line. There we go. So I hope everybody has this on their screen. And as soon as everybody has this on their screen, we can now talk about how to write a network model. And again, there's some magic under the hood, not very complicated, but I don't want to talk about that now. I would ask you to save your questions about how this little simulation works until Josh's explanation this afternoon. What we have here in this code is a place where we can type the model that we're going to run. And then in line 18, something called variables that says which variables from the model we're going to plot and what color we want them to be. And so this is a very simple code in CompuCell that allows us to take a computer simulation written in Antimony, run it and plot it. And we're going to focus today on the Antimony, the language that we use. And we're going to notice if I look at this, that it's written in a string. Here, model equals triple quotes. That's Python's way of saying that the string has carriage returns in it. And a list of the variables. So here we have variable X. Here we're going to say we're going to plot X in red. We're going to plot Y in blue. And we're going to see in line 6 and 7, something that looks like a chemical reaction. J1, the name of the reaction, colon, X transforms into Y, semicolon, rate B times X. And that's all you need to know about Antimony. This is how you write. You can write ordinary differential equations. You can write chemical reactions and so on. So why don't we actually just run this simulation? So click on Tellurium 3D, and now you can either use the project buttons to run it in Player, or here I will run it in Player myself. And I have to switch to the Player. And I may have to re-lay out my screen a little bit. And so here it's misdrawn, so I have to do my usual joggle of the corner to get it to redraw. And I hit run. And I see the time series for blue, which was chemical species Y, and the time series for red, which was chemical species X. Let's see if people were able to get that to work. Somebody says that it crashed. Are you running this, Stephen? Are you running this on NanoHub, or are you running it locally on your computer? Okay. Are you on a Linux install? Windows. That's concerning. So unfortunately, TJ has had a family emergency, and he is not available to support us this morning. So I apologize. I don't know. Somebody says it's working fine on Linux. Tony, are you on NanoHub, or are you on a local machine? Huh. I don't know what to tell you on that one, because is anybody who's running it locally having it work?
There seems to be a problem with the Windows configuration of Tellurium, so I would suggest people who's running on Windows to use it to try NanoHub for now until we can get support from TJ to fix that.
I'm puzzled because I did do this on my local install, which is a Windows install this morning, and it ran. So I apologize. I don't know how to debug that for you today. What I'd ask you to do now is no, Tellurium comes packaged with a CompuCell. So I think that I'm going to have to, I apologize, but I'm going to have to, I'm running 4.2.5 and it works. So if you're running an old version of CompuCell, it's possible there's a bug. So I would ask you, the people who seem to be having trouble seem to be relatively advanced. And so I would ask you either to, 4.2.4 is okay too. So it seems to be confined to 4.2.2. I'm afraid I'm going to have to go on then. Would I ask people to try, if you are having trouble on your local install, please try running it on Nano. And we'll come back, and everything that I'm covering now will be covered again by Josh in the afternoon. And then we can have some time to do some one-on-one debugging with you. And I apologize for not keeping everybody with me. I understand that that's frustrating. Josh, if you can reach out and try to help people in a breakout room if necessary, that would be fine. People who did get it to run, did you see the output that I'm showing here?
Professor, I tested in Windows and also in the Mac. In Mac works fine, but in Windows doesn't work.
Okay, then I'm afraid we're going to have to ask people to work on the NanoHub version for the moment.
Does it related to call the Tellurium to install it on the Windows before, or it doesn't need it?
I can't do that debug now. I'm sorry. It should install Tellurium automatically, but I can't. I don't know what the install command would be. It should be probably from Tellurium, import Tellurium as TE is my guess. Giuliano, can you look at that and see if you can see whether there's a quick fix? Again, I'm puzzled because it works fine on my 4.2.5. And so I'm going to keep going here and ask you to follow along in NanoHub if possible for the moment. And then we'll try to debug during the break. Okay, so somebody else says it works fine on 4.2.2. All right. I'm sorry that we don't have TJ here because he's the one who would be able to. Okay, so you're also 4.2.5. So it does seem to be local to 4.2.5. And my guess is, again, if you go to the top of your code, you could try typing here in line 2 it would actually be import tellurium as TE. I can't guarantee that will work, but that would be my 0th order guess for a fix. Again, I can't try it out here on the screen because I don't have the because it's already installed. It won't work. I can't reproduce that. All right. So now let's come back to the tellurium code. The things that I'm going to show you, you can mostly do. Why don't you just not worry about running it for the moment and follow along while I walk through the tellurium code, and then we'll debug it later because we have only half an hour, and I want to make sure that I introduce the basics of how tellurium works. Okay, sorry about that. Okay. So what I want you to look at now is this code between lines 4 and lines 15. As I say, if we'd been lucky, we would all have seen this 2 lines going together. Life is never so easy, I guess. And this is actually running a chemical reaction. The chemical reaction is that X is transforming to Y with first order kinetics at rate B times X. Y is being transformed back to X at a rate C times Y. If we do some simple algebra, we find out that the equilibrium ratio of X over Y is C over B. And so the first thing that we could do is a teeny exercise, and I find apologies to those of you who it's not working for, would be to change the values of B and C and see what happens. The default here would actually be C equals 0.1. Hit save. I'm going to run it again. If I run it with those values, I see X goes down much more than Y. And so I can change the equilibrium concentration. And let me come back to the code for a second. Okay. So given the time, I think I'll let you play with that on your own. But even in this very simple model, if I change the values of B and C and rerun, I will see that I'll reach a chemical equilibrium. And the value of my 2 species, the concentrations of X and Y at equilibrium, will be different. It's important to note, just as terminology, that X goes to what's called a steady state value. You'll notice that the red comes down and then reaches a constant value. That could be called an equilibrium value in chemistry and mathematics, a steady state value. The blue comes up and reaches an equilibrium value. If X and Y, if A, B, and C are the same, then my equilibrium is an equal amount of the 2 species. If my values are different, I have different equilibrium. So I want to talk a little bit now about antimony model specification. Antimony's basic idea is chemical reactions. And so you use arrows the way you do in a typical diagram in a biological network. CompuCell, and we're not going to do this right at the moment, you'll see it in the afternoon, references all of the variable names, parameters, and also the rates, E0, E1, and E2, 3 in this, are all available to you as dictionary entries. So if I define an antimony model, which turns R into Q, and I type self.sbml model square bracket Q, it will give me the value of Q. If I type self.sbml.model square bracket E0, that will give me the rate of transformation, the value K1 times S. If I type self.sbml.model and a parameter value, K1, it will give me the value of the parameter. Those can be read and written to, just like any other variable. And so I can now connect my Python level to my antimony level. I can either run the model what's called free floating. That's a single model that runs for the whole system of the model, self.sbml model, which belongs to my entire simulation, or I can load that model into individual cells, run it one version of each model in each cell.
Can we change those values during coding in the simulation, the steppables?
Yes, the whole point is that the simulation that you've written here will define initial values of the parameters and the variables, but I can set self.sbml.model variable equals 26, and that will overwrite the value in the antimony model. So yes, you can change them. You can't change the rates because those are derived quantities, but the parameters and the variables you can write freely from CompuCell level Python in the steppable. That's exactly the point of the integration.
Oh, great.
So now let's write an exceedingly simple simulation starting from this. If you can write this, you can write anything. Okay. And again, I apologize to the people who can't run it, but even if you can't run it, you can write the version here. So let's take an extremely simple model. This will be a viral infection model. We start out with T cells that are uninfected, and the rate at which they become infected is beta times T times the amount of virus, and they become I1. And so let's actually write that. I'm first going to show it to you, and then I'm going to ask you to write it. T goes to I1 at a rate beta TV. That's a rate law. How do we interpret that? We write first we put everything inside these model string triple quotes. That just tells us, and we assign that to a string variable, model string. Here, in this example, it says model instead of model string. This is just the name of a string variable. Double slash inside of an antimony string is a comment. So here, equations is just an annotation saying that I have an equation. Parameters is just an annotation saying that I have a parameter. Initial conditions is an annotation. Semicolon works like a break. It's essentially equivalent to a new line. And I can use that freely. I recommend ending every line with a semicolon. So that's the syntax so far. Double slash, comment, semicolon, break. And antimony writes everything that you do pretty much in terms of concept of chemical reactions. And so I take this diagram, which is T goes to I1 at a rate beta TV. I give that rate a name, call it equation 1, E1, colon. And then I write T goes to I1, and that's a minus sign and a caret, semicolon, the rate law, beta times T times V, semicolon. Notice that I'm destroying T and creating I1. And so that means that this one equation here implements two ordinary differential equations that are balanced. You really don't need a tellurium. There is code. Twit does have code to paste in the structure for tellurium for loading models, but all you have to write is model equals triple quote and triple quote, and then type the arrows. Okay, so our rate equation here that we want to type, and I want everybody to do this together now, instead of what we've got here at the moment, J1 and so on, we're going to type E1 equals this. Let me actually switch to this. I'll show you the result. Let's see, which one is it? Okay, this is the version we have. E1 colon T1 goes to I1 semicolon rate. And then we have to specify all the parameters. We have to specify the initial value of T here. And I've done that in two steps. I've said T0 equals 1 e to the 7th, T equals T0. I should have written probably just T equals 1 e to the 7th. It's initial value. The initial value of V and the value of beta. Of course, most things will be more complicated. I'll have multiple equations.
I'm wondering how anatomy language is kind of interpreted. So we are defining the equation first, which the beta is not defined yet. Then we assign the parameters to that, so it doesn't give an error, right?
So Antimony is not a programming language; it's a model specification. And so technically, what happens is that this string is parsed and is compiled using a JIT compiler to create an executable. This is what's called the declarative language. And I don't want to go into too much detail about it. The order in which you declare things matters only in certain respects. For equal signs, the order matters. T0 first, T equals T0 matters. It's also important that equal signs are only executed once during parsing. There are a variety of issues that will come up. I'd like to skip over the technicalities of Antimony details and refer you to the manual, because most of them are not things that we're going to run into. Antimony allows a lot of very complex modeling. Mostly we're not going to do that because we'll use the Python capabilities of CompuCell to do that. And again, we'll come to that. We will go over those issues. If we wanted to actually have a complicated chemical reaction, 3 molecules of A goes plus 5 molecules of B go to 1 molecule of A, 2 molecules of C, and 9 molecules of D. Antimony will handle stoichiometry of complex chemical reactions automatically. I can write E2 colon 3A plus 5B goes to A plus 2C plus 9D, semicolon, and here the rate is mass action, K times A cubed B to the 5th, like that. And one thing that I have to watch out for is that in Antimony, the up caret is equivalent to the 2 stars for exponentiation in Python. So the comment character and the exponentiation character are different in Antimony and Python. This single arrow diagram for chemical reaction would generate 4 ordinary differential equations, one for the concentration of A, one for the concentration of B, one for the concentration of C, and one for the concentration of D. As I mentioned, when we're going to do regulation, very often we don't care about the things that are producing the species that we're regulating. And so you can have a dangling arrow. If I have a constant source of A is produced at a constant rate A, I write that E1 colon nothing goes to A at a rate K. That means A increases linearly in time. If I have first order decay kinetics, A goes to nothing at a rate K times A, I write E2 colon A goes to nothing, semicolon rate K times A. So if you're used to ODEs, the arrow diagrams may seem a little bit, take some thinking about. If you're used to chemical reactions, writing them is very easy because you write everything in the way you're used to writing them. You can write ODEs explicitly. T accent, T apostrophe is equal to beta times T times V, creates that ODE. I can write exactly the same thing with nothing goes to T. Mathematically, it gives me the same equation. Okay. If I have the same chemical species represented multiple times, antimony will automatically sum them. So if nothing goes to T, T plus V goes to something else, then I will get nothing goes to T at rate K0 in my T equation. T plus V goes to something, destroys T, minus K1 TV. And so I can convert back and forth. Antimony will automatically sum all of the different terms involving each species. Again, it makes it very easy to build modular models, much easier than writing ODEs. And in class, I often will do an exercise where I will show a paper, for example, by John Tyson, where he's written all the chemical net reactions in terms of ODEs. And I'll ask them to say, what was the network that generated it? And you cannot go backwards from the ODEs to determine what the regulatory signaling or metabolic network was. It's not possible. You lose information. You can always go forward from the arrow diagram to the ODEs, but you can't go backwards. And so the reason that I particularly like antimony is that it's a model specification that preserves the biological information. Okay. You have to specify all of the parameters and initial values, and you do that simply with equal signs. Beta equals 6.2 e to the minus 5. V equals 10,000. T naught equals 1 e to the 7th. And here again, there's this double line where I've set T equal to T naught. That is about all you need to know about writing network models. Antimony allows you to do complicated things like events. You can say at time such and such, do this. You can say if something is bigger than something else, do that. But typically we'll do that in Python instead. However, you can do it. Okay, so what I'd like people to do now is take this code that I've got here, the one that I started out with, this one, and make this transformation. I want you to implement this viral infection model. So we started with this code here. All of these models, by the way, are available in the models directory, but just take by hand, you can go in and type, instead of J1 goes to this, we can type E1 colon T goes to I1 semicolon rate beta times T times V. Get rid of J1, J2. Okay. The parameter, there's only one parameter, beta equals, scientific notation works, 6.2 e to the minus 5. You don't need C. Initial condition. Here I've used the 2 lines. T0 equals 1 e to the 7. And then I set my value of t equal to that, T0. So why doesn't everybody try doing that?
Is this sensitive to the capital letters and low capital?
So in Python, capitalization matters a lot. Tellurium does not care about capitalization.
Okay.
However, when you use your key for your dictionary entry, it does matter. So when you move into CompuCell, you have to remember if it was capital X here, it has to be capital X here. Okay. And now why don't you figure out what you have to do to display it? It says variables. We're going to have to change my variables line. I'm going to be displaying T and I1 instead of X and Y. So everybody should be able to do that, I hope. And then as usual, you have to hit save. And now I come back to player and I'll stop and I'll hit run. And as usual, I'll have to joggle my screen a little bit. Oops, I had a typo. Global parameter V. Okay, it didn't reload the right. Okay, sorry, my mistake. I have to move it. I have to launch this now. Open the player.
You forgot V.
No, there is no V. Did I not? No, sorry. Did I? Yes, sorry. Thank you.
Included V in the equation, but you have not mentioned the value.
Thank you. I have it here and I didn't type it, right?
Yeah.
Okay, thank you. Well, so appreciate you. Also in Antimony, okay, V equals, but I have 10,000. Okay, Antimony doesn't care about, unlike Python, it doesn't care about indentation. Thank you. Let's see whether it works now. There we go. Are people able to get that to work? Looks like everybody got it to work faster than I did. Yeah, okay. And I strongly recommend using the save as inside of Twit to create new versions of things as you go. I didn't do that here. Okay. So you should have gotten something that looks like that. Okay, let's make a chemical reaction. A plus B goes to C. Why don't you try doing that? I'll do it in parallel. I want you to think about doing it on your own. I've actually got the answer here, but why don't we do that together? I gave it a new rate law name. I called it R1. A plus B goes to C. I shouldn't really talk it through because I want you to try to do it on your own. A times C. I have only one parameter, K. I set it equal to 1. And I have to set my initial values. A, I'll start with no A. Set B equals to 10. And I understand that these aren't meaningful parameters, but they're just to play with for the moment. And I have to set my initial value of C. Spacing doesn't matter. Like that. And now I'll plot A.
Should it be B and line 6? K multiply A multiplied to B. Oh,
Yeah, thank you. And C, I need to do C. I want to plot C, colon, make it red, blue, make it green. Okay. Try that. Notice nothing happens. Why is nothing happening? What was my chemical reaction?
It's A0.
A is zero.
A0.
There's no substrate, so I can't have the reaction. Nothing happens. So now let's go back, and we won't, since we're going to run out of time. I don't want to spend too much time on this. But now let's make A not 0. Let's make A initially 1. Okay, make A 1. And now given that I have to re-joggle the screen, if you use the stop and then the step function, then you can joggle the screen, get it laid out again. And now I can run it. And now you see A comes down, B comes down, C comes up. When I run out of A, my reaction stops. What if I make A equal to 10? If I make A equal to 10, do it again. Crash on me. That's a good redraw. Now I've decided to all right, try it again. Why don't you play with that a little bit while I'm getting it relaunched? Which one was I running? I was running simple chemical reaction. This was 0, so I need to open it and quit it. Okay, let's make A10 and we'll go from there. And now we come back, stop, step, layout, run. And now we have a complete reaction. All right. So you can play with that and you'll get the things that you would expect for a simple chemical reaction. Why don't you try, and this will be the last thing we'll do for this module, why don't you try implementing A goes to B at a rate proportional to A, B goes to C at a rate proportional to C, C goes to D at a rate proportional to C. Start out with no B, C, and D, and start out with some amount of A. So again, the step is write each chemical reaction as an antimony reaction, define initial values for A, B, C, and D, define the rate constants, and then put in the variables line A, B, C, and D. And let's do that as our last exercise for today. I'm sorry about the problems running Antimony on your local machines. And we'll see what we can do about that during the break. But I hope people are able to at least get this to work. Really writing reaction networks isn't any harder than this. I guess I can do it myself while we're doing it. R2 colon C colon KB CB R3 CB Cut and paste does work inside of this. So if you want to save time, you can cut and paste. Okay, I got my rate laws. A goes to B. The spaces don't matter, but it makes it look nicer if I have the spaces. A, B, A will start with 10, B will start with 0, C will start with 0, D will start with 0. And then I have to plot D as well. Orange. Did that work for people? Somebody asked to be put into a breakout room. You could help them, please. All right. So really, that's about as complicated as it gets. I hope that wasn't too intimidating. And I apologize for the bug in this 4.2.5 install for some people on Windows. But you can see now the rates of the chemical reaction. And one can play now with there are a whole series of exercises, which I give you as homework, where you can explore how these chemical reactions change as I vary the rate constants. We could make this a closed reaction, in which case I'll equilibrate. And I've got a sort of a fun model where I have bistability, autocatalysis. And the nice thing about autocatalysis is it leads to bistable states. You can explore bistability. Bistability is very important for switching behavior, where you want the cell to differentiate, or you want the cell to either decide to do something or not do it, depending on the value of an input parameter. And so those are some simulations that you can try on your own. And I'll let people try that independently. So let me do my module questionnaire. I appreciate people's patience with the issue with CompuCell. I hope that people on NanoHub, that's one reason we like the NanoHub deployment. For all of its annoyances, at least we know that if everybody's using the NanoHub deployment, everybody's having the same experience. And so I'm going to do my module questionnaire. If you want to play with the bistability model, that actually represents a gene regulatory network where you have a molecule that turns on its own production. So it's a transcription factor that is a transcription factor for itself. That model you have to interpret. This arrow to A promotes A, you have to interpret. You could do it with messenger RNA or not. but you have to think about what it means. There's a standard Hill function form for that. And you could write that Hill function form and implement it in a very simple way. Nothing goes to A promotes itself with a Hill function form. A goes to nothing, decays. And when you write those three equations in the little example we've given you, you'll find that you can do bistable gene expression. And so that's where I'm going to stop for now. So there's a question. It says CC3D suppose these processes are inside a cell. So CC3D allows you to make a choice. In the case that we're running here, we have loaded a single copy of that model to the entire system. So that model is running globally in the system. However, if I have, and I didn't create any cells in the simulation, so there are no cells to load it into. However, if I had 1,000 cells, I could run 1,000 copies of a gene regulatory network, and each gene regulatory network could be different. Each gene regulatory network could be responding to the environment. That gene regulatory network could be changing the behavior of the cell. When the behavior of the cell changes, you can load different networks. So you can actually control with very fine granularity what you're doing. You can have multiple models inside a single cell. You can load and unload models from a cell. You can change the parameters in each cell independently. You can have multiple SBML, multiple antimony models running inside each cell at the same time. You can run the models either deterministically with ODEs, as I've shown it, or stochastically with Gillespie algorithm. And you can do those. You can have some models run deterministically and some models run stochastically at the same time. There's a lot of flexibility. I wanted to show you the very simplest thing, so that I hope it didn't scare too many people away. It looks like I scared 1 or 2 people away. Yes, if you write PBPK models or other kinds of pharmacokinetic models, they're written in exactly the same way. So if I wanted to model transport with compartments, I can do that. SBML, I keep saying SBML because it converts to SBML. Antimony allows you to define compartmental models. And I can have a model of the body with organ systems, and then I could have my CompuCell be a part of the tissue like the liver and run individual metabolic models inside each cell in the liver. So yes, that's pretty common to run a PBPK model in the whole system to represent the movement of a drug or molecular species in the body, then pass that to the individual cells and have them do their metabolism or respond to the drug. So if you wanted to do a model, for example, of drug dosing for chemotherapy, you'd do it that way. Any other questions?
Professor, regarding some functions like load A and simulate that it is possible to work with from the Python. So I don't see that it is used here in the code. So how does CompuCell works with that?
That's exactly what I said. I asked you to not ask that question. I'm teasing you with it because the whole afternoon, Josh will be teaching you how to do that.
Oh, okay.
We created this template model so that people could just write the antimony and see it display, and not have to worry about loading and compiling things. So in the afternoon, we will show people all of the details about how to write these models, load them, change the settings of the integrators and everything else. But I wanted to give people something where they could immediately say, I have the reaction, molecule A plus molecule B gives me C. How do I get it to work in CompuCell? And so that was the focus today. It's a very good question, and it's exactly what we're going to do later. Okay, any other questions before we break? We're running a little bit late. Why don't we come back at, let's see, we were supposed to have stopped at 12 o'clock. It's now 12:10. Why don't we take a 10-minute break, and can we start back at 12:20? Is that all right for people? And for 12:20, we're going to be doing the hackathon discussion. So if people have previously did hackathon presentations, if you're willing to give us a little update on what you're thinking about your hackathon ideas. If you're somebody looking for a hackathon team to join, please speak up. And I hope that the people who were not able to speak on Tuesday are going to be ready to present their ideas. Okay, so I'm going to, I hope that people can, maybe during the hackathon session, we can have people work trying to make sure that everybody can run this. If we cannot get the 4.2.5 issue solved, and if TJ is not on, I'm not sure we're going to be able to do that. If we can't, yes, Giuliano, you have a solution?
I did some debugging. Yeah, it seems to be that Antimony is missing from the 4.2.5 installer again. But in 4.2.4, it's there. So I recommend people installing CompuCell 4.2.4 as well. I can grab the link for that, but
Okay,
keep it as a separate installation. As MaBoSS is present in 4.2.5 and not 4.2.4.
Okay, so I will see you in a few minutes after the break. Okay, welcome back, everybody. This session is devoted to discussing the hackathon. And I wanted to first give the opportunity for the people who were interested in participating but weren't able to present Tuesday to be able to present their material. We still have some people, we still have a number of people who are unsure about whether they want to participate. And we have at least 7 people, actually 13 people who are potentially looking to present their ideas. And so if you have your slide queued up, that's fine. If you don't, speak up when we get to you and tell us what you're thinking about, okay? So I need to remind you again that this is being recorded and live streamed. So please be aware of that. And I wanted to, actually, we'll flip the order. We'll do the additional talks first. And then we'll walk through, what I'd like to do is walk through the peoples who presented on Tuesday and give them an opportunity to say whether they made any progress, what they're looking for, and anything else that we could do. We could create some additional breakout rooms if people would like to have a breakout room to chat with possible collaborators and this kind of thing. Please let me know. We can try to set that up for you. Put it in the chat. And when we're done with our discussion, we will have Josh take over and continue the presentation of the Antimony models, and hopefully answer some of the questions that were being asked at the end of the last module. My only concern is that I do not see Josh on the call. I hope he is not having internet problems. He's coming to us from Puerto Rico, and so I hope that he's not having internet problems. If he is, we will improvise. So, reminder, we have the folder. Giuliano, if you could repost for me the hackathon single slides folder for uploading your ideas. You're welcome to make those slide decks more detailed as you develop your hackathon ideas. We also have that hackathon ideas partnering document, which people have been using. And there is the hackathon Slack channel. So if Giuliano, if you could repost those links, that would be helpful, or I can do it as well. Okay, let me do it. So I think Giuliano may be trying to debug this issue with, okay. That was the slide deck posting. This is the Google Doc, and let's see where did the hackathon channel go? So that should be all the links that you need for the moment. So I'd like to do the additional lightning talks. I think one person who was going to do this, was interested in doing this, just texted me and said they're not able to do it. Let me see if the people who did ask to do it are there. I see at least one of them is there. So why don't we start out with David? If you're there, why don't you tell us a little bit about your idea and make a little pitch if you'd like to have some collaborators.
Thank you very much. My name is David Fagbemi, and I will be discussing my project. So what I'm trying to do is I'm trying to model a cellular signaling pathway of human bronchial epithelial cells. So epithelial cells are the cells that line the inner coating of the bronchial of our bronchial system in the lower part of the lungs. It's what protects against respiratory viruses and other, external actors. So that includes things like allergens or smoke or whatever it is. So since all these foreign objects create a certain immunological response, like the release of cytokines and chemokines, we're trying to figure out how to predict cellular signaling response based on each and every one of those viruses. So we came up, what I'm using right now is a basic, a logic-based model, which is kind of like our base, which you kind of create a basal or a fundamental baseline model that will be able to show us, okay, how this is what happens when this virus comes into the system, and how we're going to be able to sort of, understand the pathway signaling system, because that's the most important thing we're trying to figure out, like how to understand the signaling pathway systems so that we can be able to, better design drugs to be able to affect each signaling pathway. What we use right now is NetFlux, which is kind of like an open source software based on MATLAB. It allows us to model these pathways. So I was trying to find out if there's any other tools that we can use in terms of in a spatial temporal context. For example, something like CC3D, which we mostly focus on which focuses mostly on spatial modeling. So I'm trying to find out if there's any other methods or models that we can use to be able to pretty much model this pathway. Yeah.
Okay, that's great. We definitely can come back to that. There are some tools. The problem with, I don't know NetFlux.
Yes.
The problem with writing MATLAB code is that it's not shareable, and it's not usually convertible. But there are some software tools that may allow you to convert, depending on how elaborate your models are. Recoding them in Antimony may or may not be practical. If you have 50 equations, recoding them in Antimony takes an afternoon. If you have 2,000 equations, recoding them in Antimony takes a long time. But in principle, these kinds of models could be expressed using the language that we just showed today. If there's an exporter from NetFlux to SBML, then the models can be read and written directly in CompuCell. And if it's exported to SBML, it can also be turned in Antimony if you want to be able to edit it. So those are things that can be explored. Okay. But that sounds like a great project. So we can definitely talk more about that.
Can I show you like an example? Can I share screen and show you an example of a signaling pathway, just to give you an understanding of?
Sure.
Okay, sure. Here. Give me a second. Yeah, so this is an example of a signaling pathway here. Let me zoom it right in. So you have the receptor levels. So there's multiple stimuli. There's multiple stimuli that can be used, and we can see each. Let me shrink this. Okay. So previously, what has been used to model a system like this is we mostly be focused on gene expression. But we realize that gene expression is like the final phase of a cell signaling pathway. So we're trying to go through the entire cell signaling pathways to sort of understand, we're trying to understand, okay, the entire network. And if we can understand the entire network, we can be able to predict cellular response. So that's pretty much what we've focused on. So here you can see our stimulus, our input level, which is the stimulus level. Then we see the receptors. Then we have the adapter molecules. all the way down to the release of our cytokines down here. Hold on, give me a second.
And is this a Boolean model then? Is
Yes,
this a
so it's a-
Boolean stochastic model?
Yes, so we use a mixture of an ODE and a Boolean model. So we call it a Boolean-based or a logic-based differential equation model. So we try to combine both of them because both of them, as good as they are, they both have their strengths and their weaknesses. Boolean models are more scalable for a large network like this, but it doesn't give us this specific, it doesn't give us a lot of mechanistic insight or quantitative. In terms of accuracy, that's what ODEs do. But ODEs cannot really be applied to like a very massive like network like this. I mean, we can use them, but it's so cumbersome. So that's why we try to combine both of them. We use the scalability of a Boolean model and we use an ODE to give us much more accurate, understanding of the dynamics of this cellular signaling pathway.
Okay, that's so CompuCell now supports Boolean models through MaBoSS. So if we could translate your code here into MaBoSS, then it can run. And the ODE models are supported through SBML, CellML, or Antimony. So in principle, we could do this. Of course, the amount of effort to translate, it will depend on, we'll have to look at it, but it's definitely a reasonable thing to think about. That's a great project.
Thank you, sir.
And seeing that in a spatial context would be fascinating. So let's go on through the other talks and then we'll come back, circle back and let people discuss more. So that's great. Thank you.
Thank you, sir.
The next talk was from Hamed. Is Hamed here? Let me look at our participant list, see if they're here. Okay, I do not see Hamed here now, so we'll go on. The next talk was from Marco Rusconi. Are you here?
Yes. Can you hear me?
Sure, please.
Okay. So thank you very much, first of all. This is an amazing workshop. And my name is Marco Rusconi, and I'm a PhD student at Institute Curie. For the hackathon, I'm proposing this model, this 3D model of cancer invasion, which include an intercellular signal, intercellular signaling, this Boolean network. I already developed this model with PhysiBoss, which comes from the merge of Multicell and MaBoSS. And so the objective of this model would be to simulate the different modes of cancer cell invasion through the extracellular matrix. So to monitor single invasion, collective migration, and trail migration, while also considering the formation of invadopodia or lamellipodia. So the goal would be the goal of this model would be to actually introduce some mutation in the Boolean network and see the actual results on the simulation. Like for example, what I did with PhysiBoss was to mutate one of the node of the Boolean network with MaBoSS and completely block or force migration in the simulation in order to reproduce an in vitro experiment from Lodilinski et al. And also in the future, I would like to, since this Boolean model is still a work in progress, I would like to, in the future, to include new pathways to also reproduce these experiments where lamellipodia are activated through stimulation with blue light that activate optosarc. So that's it basically. And I already developed this model, and I have here, if I can show the screen, I would like to show a 3D example of the model. Here, I don't know if you can see it. So I would like to reproduce something like this with CompuCell 3D. The white cubes are basically extracellular matrix, and so the cells would degrade and escape through the extracellular matrix. And the microenvironment would be composed by extracellular matrix, TGF beta, and oxygen. And those would be also the input of the Boolean network.
Right. Well, that, I mean, the only thing, how many cells are in that simulation?
So in this simulation, I started with, if I remember correctly, something like 500 cells, but maybe this is a bit too much.
No, that's fine. For 500 cells, it's no problem. So I don't know if you were on yesterday because of I know the time zones are a problem for Europe. But when Josh showed this, the simulation, the chemotaxis simulation, where the cell consumes ECM and then migrates in response to the gradient it creates.
Yeah,
that's essentially exactly this simulation.
Yeah, exactly. In fact, I was super interested in that.
And now that we have the MaBoSS integration, assuming it works, because I haven't used MaBoSS, so we need TJ to do that. But in principle, converting this model over, there always are problems. My instinct is it's a 2-hour job to convert it.
Mm-hmm.
In practice, it's probably 2 days.
Okay.
Because there are always bugs. But doing this conversion should be straightforward. If the MaBoSS model works, it's fine. The cell ECM interaction is fine. I don't see any problem. That's a great problem. I would love to see that implemented. So I hope some people will decide to join you. If not, we'll be there to do our best to support you in that. So that's great. Thank you very much.
Thanks to you. Thanks to you.
Okay. And then the next one, Suvik here.
Yeah. So, okay. So let me share my slide.
Okay. Well, I should have. So do I not have the?
It's saying that you cannot start a screen share. Okay.
Was this a movie or is it a static image?
It's a static image, yes.
Okay, so tell us what your idea is.
Okay. So before coming to this work, so I don't have much idea about the CompuCell 3D, but so I have learned a lot from this workshop, and thanks to organizer and also the participant for this help. And so I will be studying a very simple model. So it's basically I want to study the confluent cell monolayer with cell division and apoptosis, and I want to characterize the glassy dynamics within this type of model. So the motivation behind this work is basically the collective motion of cell in a confluent monolayer is important in morphogenesis, cancer metastasis, wound healing, and etc. So recent studies and experiments have shown that the dynamics in such cellular system has remarkable similarities with that of a glassy system. So by the word glassy dynamics, it refers to the extreme slowing down of the order of like 10 to 12 order of magnitude with a small change in control parameter. And so that glass transition occur without any reasonable signature or reasonable change in the structure or any phase transition. So in this project, I will be in the context of the active matter. What I will do, so I'm interested to see whether this glass transition can be found in the cellular POTS model using CompuCell 3D, and in presence of this cell division and apoptosis, which is the cell cycle events. And I will also like to study how the rates of the cell division and apoptosis affect the glass transition. And also, if time permits, I will also like to study the active polar cell to characterize the glassiness in this type of polar cellular model. So, and also I'll also like to see that if there is any lattice type effects or not. I hope that there will be no, but that I will like to check. So, and that's the plan of my study in this hackathon project. Thank you.
Okay. I realized that what was covered yesterday by Gilberto went by a little fast for some people, and I definitely, but Gilberto's done a lot of work on the statistics of tracking cells. And so I think a lot of the how determining the trajectory of the cell, calculating mean squared displacements, these things are things that he's an expert in. So if we could get him involved.
So I have a short discussion with him in the Zoom chat box. So that helps a lot. Actually.
Right. Great. So that's
and also Professor James Lucas helped me in some part.
Good. Well, I think you're joining in the middle of the night, so I appreciate your staying up at night to be part of this.
Thank you.
Okay, great. Okay, so those were our new presentations. So now I'd like to run through the list of presentations that were already made and give people the opportunity to update people. If you don't want to update, that's fine. Just say that you don't have anything to say, or if you're not here, of course. I guess what's the young Gara comment that if people don't want to go to the ball game, nobody can stop them. So if they're not here, it's going to be hard for them to speak. So, but if you'd like to do an update, we can do that. If you would like us to create a breakout room so you could have discussions with team members, that's fine. Please let us know, post in the chat, and we'll do that for you. The one problem that we have with Zoom when it's set up the way it is that I cannot leave the main session. So the person who owns the Zoom session has to stay in the main session. And so I am restricted. I'm happy to talk to people one-on-one, but I'm restricted to staying in the highest level of the Zoom. I can't join the breakout room. But if people would like to have a breakout room, that would be fine. So I'll just run through these, and if people see their names up on the screen and they're here and you'd like to update us, please let us know. If I don't hear from you, I will go on to the next one in just a second.
Professor, can I share my screen?
Please, go ahead.
Do you have my screen?
Yeah, that sounds
good.
Yeah, the project was about the mechanobiology of endothelial to mesenchymal transformation or the calcific aortic valve disease. In this project, we possibly try to apply some shear force on the endothelial cells and some maybe chemicals or cytokine or whatever. And in order to see how the endothelial cells kind of triggered to secrete some chemicals in order to degrade ECM and go inside the ECM and make this transition. So just as preliminary test with the computer cell, I made some modeling, but it is not, I was just playing with the things that we have learned up to now. So here, the red cells are the endothelial cells that I put. The yellow one is the base membrane layer, and the green ones I modeled as the ECM. What I did here, I put some cells like the blue ones in order to secrete. And once the secretion of some chemical field reaches to the endothelial cells, then they triggered and they start secrete another chemical in order to degrade the ECM, and using chemotaxis, they try to dig into the ECM.
So that actually sounds a little bit like the proposal that we just heard for the cancer model from Marco, right? With the degradation.
Yeah, here I use the adhesion flux for the energy. I mean, I defined some cadherins and integrins, but I need to adjust the parameters for them because for the chemotaxis, they build up on top of each other. I mean, the red cells. And we go like this. So I was thinking to divide the cells, endothelial cells, for different types. And when they are activated, I apply chemotaxis on the activated ones to not get other cells involved in the chemotaxis. So I am trying to figure out how to apply chemical force on top of these endothelial cells. And also, this would be kind of multi-scale modeling. So for the endothelial cells, I'm trying to build with the tellurium subcellular model. That's it.
Well, that's really impressive. One thing that sometimes happens with chemotaxis is that to get the chemotaxis forces to work properly, in a lot of systems there's adaptation, and in that situation, it's easier. What you could do is you can adjust the strength of the chemotaxis parameter cell by cell in response to the level of the concentration. So you essentially make it a logarithmic chemotaxis. That should really be a built-in function in CompuCell, but you have to do it by hand. But you scan the cell, you find the mean value of the chemical field for the cell, and you divide your chemotaxis parameter by that. That allows to have a much stronger chemotaxis response. But this is impressive. I hope it will inspire. I hope it will inspire people. I think it's a great.
Thank you. And also, I'm thinking that, for example, when the cells, the endothelial cells are activated and they secrete some chemicals, then other cells follow them because of that secretion. But I end up seeing that they are kind of bind them together and go another way, like fly. So other ways because of their mutual interactions that trigger each other.
Yeah. So that's something we can. Yeah, the chemotaxis is complex. You get there are a lot of, because cells respond very differently. Some cells only measure the chemical concentration at their leading edge. Some respond in different ways, and so there are a lot of options to consider. Again. One of the things that we did get to, it was already complicated enough yesterday, was the model Gilberto showed. You could bias that with chemotaxis and then have those cells crawl up a chemical gradient. So there's a lot to do. That's really impressive.
Thank you.
So I hope are you looking for people to work with you, or do you want to just do you want to do this on your own?
Actually, 3 people told me to join with Dr. Tion West, I think, one, the Narshin Fatima, they are not here, I think, and also Noah Ishkar. So if somebody else want to join, it would be appreciated.
Okay, great. I will create a breakout room for you just in case. The one problem with the breakout rooms is I have to create them all at once. I can't add a breakout room while they're open. So I apologize if I have to wait a second for doing that. But that's wonderful. That's impressive. Let me go back to the screen share. Okay, the next talk was AJ, and did you have anything to update? Comments?
I don't have an update really. I guess my plan was to work with Abhishek, who's a postdoc in my lab.
Right.
And I haven't heard from anybody, but I'm okay with working with Abhishek. But I would, if somebody wants to join in, they're welcome.
Okay. So I guess the one problem with the I'll put the list of projects back on the screen at the end, because if people want to join breakout rooms and they're named after the speaker rather than the topic, you have to remember who's doing what. Okay, great. Farnoosh, do you have an update?
Can you hear me? Hi. Yes, I have something to say. The title of our project, as you see here, is Modeling the Immune Response Following Monogenic Cell Death in PDAC. I was lucky to have 3 teammates so far. Madish, Tahrim, and Paula have joined. And now we are a diverse team with teammates from all around the world, and with diverse expertise, with biology background, modeling experience, and Python skills. But we are all beginners in CC3D, and as we have still 1 or 2 places on the board, we would be happy to have a 1 or 2 new teammates, especially with CC3D experience. Today, we had a meeting together, and it was very great experience, and I think you will have fun during the hackathon. I already talked with TJ, and he had some piece of advices. He also told that probably Joshua can help us, and Joshua kindly accepted to have a meeting with us probably today after the workshop or tomorrow before the workshop.
Wonderful. I'm really pleased to see people getting things organized. I'll create a breakout room for you. Again, these aren't, the Zooms are not the most efficient way of doing these kinds of organization, but at least we have some. Okay. Sorry, go ahead. Were you not, did you have something else you wanted to say, Farnoosh?
I don't know if it is possible for Joshua to have a breakout room now. I don't know, because he was busy today. So
he's here.
Yeah, I'm available.
Okay. So the only thing is the only problem is I can't create the breakout rooms. I can't add to the breakout rooms while they're open. That's the only problem. So give me, let's spend just a few more minutes going through the list, and then I'll open all the breakout rooms and people can organize, okay? If that's all right. I don't think it should take too long. Megan, you have an update. I know you said you have a good team put together.
Yeah, can I share my screen? Okay, can everyone see my screen? Great. So just a quick update. I have a proposed team. So me, Tian, Alexa, and Aaron, but we're still open for anyone else who's interested in joining. We've kind of fine-tuned our goal a little bit. So we want to develop an agent-based model of skeletal muscle that predicts the dynamic regenerative response of muscle cells, fibroblasts, satellite stem cells, inflammatory cells as a result of an acute injury, and also potentially look at varying levels of estrogen. So our specific modeling questions include: how does estrogen alter the outcomes of muscle regeneration? Do spatial inflammatory cells and growth factors change the injury recovery dynamics? Comparing that to a previous model that we have that does not have spatial aspects for the inflammatory cells and growth factors. And also looking at the initial conditions and which of those have a major influence on muscle recovery. And then at the bottom, I included a screenshot of kind of what we would envision the model to maybe look like. So importing some type of muscle histology, and then you would see the damage and the cells move around and help regenerate the muscle fiber.
Wonderful, thank you. So, Yajun, is it?
Hi, I have nothing to update, but I'm still welcome if anyone is interested in this neurons migration modeling, and it's also okay if I just do it by myself.
Okay. All right. Aaron, are you working with Megan? Is that right?
Hi, yes, I am going to be working with Megan. I did also get a message from Kato. Sorry if I'm mispronouncing that, but I didn't hear back from her. So I don't know if she wants to work on something on this in general, but I can hear from her independently if that works.
I just want to make sure that if people want to I'm sorry again, this is always a little bit klutzy with the all right. Janos or Janos?
Yeah, I'm here, thank you. I updated the slide deck with one slide about my previous ideas. Which
so
is
you want to show a new slide?
Yeah, I can share my screen. Maybe. Okay. How do you?
It's starting.
It's starting. Okay. Do you see my screen right now?
Not yet.
Not yet. Okay, it's it's a bit slow. Okay, so what's the problem? Okay, is it visible or not?
Maybe no. Let me take the screen share back and
okay, and I updated at the slide deck these new slides, so you can also show it if it's easier that way.
Let me see if I can find it. Give me 1 second here. There it is. Okay, does that work?
For me, it's still frozen somehow, the screen.
Okay, for some reason. All right, is that working for people or not? Okay, can you try to stop screen sharing?
Actually, I can see the PowerPoint on my phone, but I can see it on my laptop. I don't know why.
All right. I'm sorry. Maybe,
okay, now I see it.
Okay. Go ahead.
Okay, now I see. Yes. Is the new information on the second slide that I thought that the tumor model from yesterday, it's a bit similar to what I'm trying to do. It's on the second slide because it has two cells, one that divides at the edge because it has nutrient and so on, and at the center there are cells that not divide anymore because they don't have enough nutrient or oxygen or something like that. So, but it's a three-dimensional colony, so some kind of gravity and this gravity would be needed, and also attachment between cells. I don't know which is the easier, making a three-dimensional model at first or a two-dimensional slice, like in the images. microscopic image? Is it what is the easier to implement in the system?
Well, I guarantee a
was
2D
one of my
model is easier to start with. The 3D models sometimes the 3D is essential. So when you need 3D, you should do 3D. But they're bigger models, they run slower, and they're a little more complicated. So I would start with 2D. Julio will show a buckling model, epithelial buckling model tomorrow. And if you combine that epithelial buckling model with diffusion limited growth, I think you could probably replicate this pretty well.
Okay, then it's great. It's great. I already talked about with Tahrin, but I'm open to have other group members to work with, because I'm new in CCD. I already did modeling in MATLAB, but it's the computing language is quite different. But these images show how the model should look like. Yes, how the result should look like qualitatively. So I would like to get something similar to that.
I think that's quite reasonable. So I've created a breakout room. Again, I apologize to everybody. I can't create the breakout rooms until we've done with this section, because I have to do them all at once. So let's quickly go through the list of updates and people who would like the breakout rooms, and then we will together. Great. Okay, is Kafyan Atari?
Yeah.
Here?
Hello all. Well, actually, I don't have much of an update, but Giuliano agreed to give me a hand during the hackathon days. So again, if someone is still interested in this project, I am open. So that's all.
Okay. All right. Maria? Maria here? No. Okay. Lorenzo, thank you for all your help.
No worries. Well, I don't have big updates. In general, the project is still on looking at Notch and trying to extract some information and modeling that from data and modeling that. And essentially, I think the task will be implementing proliferation in a pre-existing base of model, and perhaps the most interesting bit is trying to set up a parameter scan and try to see how we can convert the data back and cross-analyze them with our experimental data. So I'm happy to have anybody on board. If anybody wants to or is interested, I'm open. So.
Okay, so I'll create a breakout room for you. So is there anybody who has not spoken so far, who is interested in proposing a topic?
Hi, yeah, I have a project I'd like to propose, if that's okay.
Okay, I'm sorry, can you introduce yourself? You
yeah,
need to screen share.
I'm Jonas. I did upload it in the Google Drive, but it was kind of just a few hours before the course, so I can just share my screen if that's easier.
Okay, sure.
Let's see. All right, can you see this? Can you guys see this? Oh, sorry. Yep. So the project that I'm going to try and do is to model the effect of mitotic count and tumor infiltrating lymphocytes on the radiosensitivity of HPV-associated oropharyngeal squamous cell carcinomas. So just a bit of introduction about it. These human papillomavirus OPSCCs, they tend to have a pretty good prognosis and respond quite well to radiotherapy. And also another thing is that they these patients with with high numbers of lymphocytes in the tumor immune microenvironment, they also tend to have good
Kostabi?
outcomes.
At some point,
And
I
one
should
thing
get
that
my mum.
one
Because
thing
you
that
have
we
to be there by 2:30.
Yeah, I have a 1:30 call, so
hello.
Ajay needs to mute. Yes, thanks.
So should I continue?
Yes, of course.
Oh, yeah. Okay, sorry. Yeah, so I was saying the one thing we also know is that high mitotic rates of this tumor cell is one of the reasons that has been suggested for its radio sensitivity. But of course, with a lot of other tumors, we know that if a tumor has a lot of mitotic counts, they tend to be quite aggressive and have poor prognosis. But I think in this case, the pathologists are not very sure as to whether or not high mitotic rates are a good or bad thing in these tumors. So what we have is actually H&E biopsies that we have processed through an image analysis workflow, and you can see that we have the outputs are the single cell maps of the tumor cells, as well as the lymphocytes, which are here. We also do have information about which cells are undergoing mitosis at this sort of snapshot in time, and also which cells, which lymphocytes have infiltrated into the tumor islands. And so the idea that I'm going to try and propose is to develop really two basic models. One is to model the effect of mitotic count versus the aggressiveness of the tumor and the radiosensitivity of the tumor. And the other would be to try and model the infiltration of lymphocytes from the stroma into the tumor islands in a sort of chemoattractant or chemorepulsant type of situation. Yep, and I'm really happy to have anyone, if anyone's interested in this project. That's all. Thanks very much.
So I think that's everybody. Appreciate your so I think I have everybody's down as a breakout room. I will open the breakout rooms, and people are free to join and go in as you see fit. Please let me know if you have any problems. I've tried to give the breakout rooms names that are at least partly descriptive of what people describe. Everyone should feel free to join in. If people want to talk to me, as I say, I need to stay up at the top level just because of the way this is structured. We have breakout rooms from Megan Haas on skeletal muscle, Ajay on lateral line, Javid on tissue invasion, Farnoosh on immune response, Ayajun on neuron migration, Aaron on zebrafish epibole, Janos on East colonies, Imam on cartilage, Lorenzo on delta notch, Jonas on mitosis and radiation, David on bronchial signaling, Marco on cancer invasion, and Suvik on cancer, and Suvik on glassy epithelia. And for those of you who are not interested in joining a team, we can chat, or you can take a coffee break, and we'll let people have a conversation. Again, I appreciate people's patience with the running a Zoom meeting. But I think they're wonderful topics. I'm really pleased that and some years the topics that have been proposed are not good matches to CompuCell. I think every one of the topics that's been proposed is achievable. Whether we can achieve it in 2 days is another question, but achievable. And I do appreciate people's patience for this bug in 4.2.5 with tellurium. So feel free to unmute and speak up on any topics that you find useful. This is just a list of the speakers from previous. If anybody needs help moving into a breakout room, you should be able to move on your own. But if you want to move into a breakout room and you can't do it, put it in the chat, and we'll go from there. Well, the next session depends a little bit. The next section will start at, let's see. In principle, it should be 2:15. Is that right? Gosh. And again, free to have a conversation now while we're discussing. TJ, are you there?
Yes, I'm here.
So there's a problem. Antimony tellurium doesn't seem to work in 4.2.5 in the Windows install for some people.
Really?
Really. It works in mine, but it throws an error. So if anybody who has had this trouble this morning and that would like to explain the error, that would be a good opportunity.
I think that in the latest hotfix, Antimony got kicked out of the binary installer for some reason.
So is that just a problem on NanoHub?
Windows.
NanoHub works.
Yep.
And again, people are free. I just wanted to let the TJ know about this issue. Somebody just put this. Okay. Thank you for sharing the error message, Joseph.
A question of the hackathon.
Yes, please go ahead.
As an observer, I may be curious to go from one break room to the other.
Yes.
And not promising to contribute anything, but just out of interest. Is that possible?
Of course.
So
I mean, I suppose the team,
breakup rooms will be advertised then?
Well, the breakout rooms at the moment are just for this next half an hour.
I'm clear about that. I'm asking about Saturday, Sunday.
So for Saturday, Sunday, you're welcome to come. And I mean, I suppose if the team says they don't want an observer, they can say that. But the plan is that I'm sure that people would be happy to have somebody to discuss with, even if you're not a main participant. But the organization.
There will be a list of breakup rooms that I can move.
We'll do our best. Yes.
Yeah. Okay. Appreciate it. Thanks very much.
So concerning the bug, there is a way that I can walk Windows users through for fixing that. I don't know what that does for us, but it's a pretty simple fix. I don't know how this happened, but yeah, we can fix it with should be one command in the terminal.
If anybody wants to move to a breakout room and is having trouble, either speak up vocally or put it in the chat and I'll move you. So TJ, do you think that there's a fix we could send out to people so that when Josh starts his presentation, we can what people can run?
Yes,
Okay.
I can write up the instructions to send out via email and post in the Slack. And then can walk people through it as well whenever necessary.
So, Iman, I sent you an invite to the breakout room. You should be able to join there, but yeah, okay, you're there. I see a lot of people who are alone in their breakout rooms. So I don't know how efficient this is. I mean, it may be that it might not be a bad idea for people for instructors to migrate and check out the breakout rooms if possible. If you have the fix, perhaps we should make that an announcement for the group.
Okay, I'll write up the instructions now.
I know things have been complicated today, so I appreciate it.
Yeah, no problem. Sorry this happened. Again, I'm not sure how this happened because this is not something that typically changes in our binaries.
Well, I was surprised because I was working yesterday. I mean, it works for me for the.
And James, have you installed the 4.2.5 binaries?
Yes.
And you have a Windows, but other people aren't getting it. That's.
But it seems to be the Mac and the Linux people didn't have a problem, and it works fine on NanoHub.
Okay. I'll talk with Matt about that. That's, yeah, that's very peculiar.
Does anybody need to have be moved to a breakout room? Want to discuss anything? Or we have about 20 minutes now to have free discussion.
I actually let's talk a little bit about the project that I'm working on. I'm not going to be there for the hackathon this weekend, so that's why I have been involved with that. But yeah, I could do that here or in a breakout room with someone. It's a very simple project in a sense that we're just trying to smush some cells to recreate some shapes that we're seeing. And in a way, it's kind of misusing CompuCell 3D because they're not actually even cells that we're modeling. But CompuCell 3D was something that allowed us to change the shape while retaining the volume very easily. But let's talk to them about how to get, because what we're doing is starting with spheres and then wanting them to deform by kind of smushing into each other and line with each other. And so I did that initially by just trying to make some contact energy between the cells so that they like zip up together. And it somewhat works, but the problem is that they have too much of a tendency for the membranes to just kind of get all variegated to maximize that energy by interdigitating a bunch, which is not what we want at all. We really want the whole structure to kind of smush up against each other and eliminate any space between them. So I'm not sure if like some sort of force or gradient or something would be best, or.
Well, so with the cell models, typically what you do then is you constrain the. Did I lose my internet connection?
No, yeah.
Okay. Did you constrain the surface area of the cell separately from the contact area energy? And so if you want to use a negative contact energy for axial adhesion, then if you don't constrain the surface, the cell will create an infinite amount of contact energy and basically turn to dust.
Mm-hmm.
But if you constrain the surface area separately, then you can define the surface to volume ratio of the domain of the object, and have the adhesion do the things that you want. It tends to be a little finicky in terms of parameter choices. You have to fine tune it a little bit, but that does tend to work. But I don't know if you have a simulation you want to show quickly while we're discussing.
Sure. Or maybe what I can show you is some like screenshots. I'm trying to think. It runs slowly because it's a 3D simulation. Right. Yeah, let me just a moment and I'll see if I can grab some screenshots that I've saved. I don't know if Garrett is here. I have a student.
I'm here.
Yeah, if you have any screenshots, Garrett, just kind of like a final.
Oh, I have a ton of them.
Yeah, you do. That's what I was thinking. Then maybe you'd like to. Yeah, maybe I'll let you do that, Garrett. If you could share a screenshot too of what it.
PJ, I think Marco would like to talk to you about the MaBoSS project, but probably you should send him a message about that.
Okay. Do I?
Or if you want to join his breakout room, I think he's sitting there waiting for you.
Oh, yeah. Who did you say was again? I'm sorry.
Marco Ruscone he's a MaBoSS person.
Okay, so this is one of the ones we've been working on.
So again, these start as spheres, right. And then after 1,000 steps or whatever, right. Or 600 steps, it looks like they did smush together, but they also the boundary is getting all messed up.
So yes, so then depending on the physics of what you're trying to model, it may or may not be true that the surface area is constrained. If it is, then that should help.
Okay. No, I mean, of course, just smushing a sphere to anything that's not a sphere also increases the surface area, but I suppose less so than all this pixelation, so it should still help. Huh?
Right. Okay. So if you add a surface area constraint, that should help a lot. The one thing you have to do is if you're changing the target volumes, then you have to adjust the target surface to match.
Right.
So one thing that happens is typically when we grow cells in a tumor simulation or something like this, we adjust the target volume, and then you have to. But if you don't adjust the target surface area to match that, you'll get it's like blowing air into a beach ball. It'll get stiff. Right. And so you have to then do usually a back of the envelope calculation where you say, if it were a sphere, how much surface area would I have? And then you adjust that, you calculate the effective radius and you adjust the two parameters together. So you change the volume, the surface area to be like the volume to the 2/3.
Yeah. We're not expecting to change the volume, so that should be okay. They should just stay with the volume they started.
That was actually a mistake that was made very long time ago, back in 1992, where the surface energies, we just, by default, the contact energies were defined to be positive on the assumption that if the energy differences were right, the actual value didn't matter. And in fact, things like the diffusion constants of cells actually are different. If you have a negative contact energy, you have a higher diffusion constant than a positive contact energy. Again, because of this fact that a non-existent unit of surface has an energy of 0. And so it's in most physics, of course, only energy differences matter. And the only energies you've written down are the ones in your contact. But in fact, there's always a 0 there, whether you've written it or not. And so going to between positive and negative contact energies actually does change the simulation. because you can destroy surface. And that's a trick that we run into a bit. Okay. Does anybody else want to join? Anybody else want to join a breakout discussion or have any issues they want to discuss at all? We're a little more than halfway through the course, and I know not everything has worked, so I appreciate your patience with that. I appreciate you coming back despite that. So any comments or ideas that people want to bring up, please let us know.
Hello, Professor Glazer. I am Haplia Ioan from Romania. I would like to ask you something about not so technical, rather principle based. Would it be possible to change the relationship between the system being modeled, a biological system, and a model like CompuCell, to consider CompuCell itself as the object, the ideal object, and to replicate some parts of its behavior with simple ODEs. So we consider the spatial model as fixed and ready-made, and then try to find some parameters for an ODE that captures some parts of the system. Do you think it would be a fruitful direction or not?
So that's a fascinating question. Over the years, and I can't tell you how many years, it's 25 years maybe, Yanis Kavrakidis has repeatedly proposed that we should work on that problem. And somehow we never do it. So I think it's very interesting because these kinds of spatial models are very expensive. They take a lot of computing. And sometimes the spatiality is critical and you need it. And there are other times when the result could be, for example, if all you care about is the volume of a tumor versus time, a population model may be sufficient to give you that information with a few ODEs. And so we mentioned briefly, but didn't really go into it. We just had a paper accepted on this that TJ has worked on, going the other way, where we start with ODE models and we turn them into spatial models, where we map the components, of the model very carefully, so that the parameter values in the ODE model are pulled in, and in the well-mixed case, give you identical results to the ODEs. And then you can ask the question, if there is spatial variation, for example, in a viral infection model, if not all the cells are exposed to the same level of virus, because there's diffusion of virus or diffusion of interferon, then the ODE model will not agree with the spatial model anymore. And in that case, you can ask, how are they different? And one of the things that you find out, for example, is that if you calculate the infectivity of the virus, which is that beta parameter that people use, if you assume an ODE model, which is a well-mixed model, then, and you actually do fit the curves for infection to the experimental data, you'll misestimate the infectivity because of the neglect of the spatiality. And so we've worked on the going from ODE to spatial models. Having formal ways to understand how to go backwards would be very, very helpful. And that's, I think, quite a profound question. The other thing people have thought about is whether you can use artificial intelligence methods to do what we would call surrogates, where you would, what you're proposing basically are ODE surrogates for particular whole models or components. For example, diffusion equation is very expensive to solve computationally. And so for big models, very often most of the computer cost is actually solving the diffusion equation. And so we've been doing some work trying to develop artificial intelligence. That's a fancy word. Neural network simulation. I don't like the word artificial intelligence, but people use it. Neural network simulations that say if you have a configuration of sources and sinks, what is the equilibrium chemical field? And if you can do that with a neural network, it runs about 1,000 times faster, even 10,000 times faster than actually doing the computation directly. And so the problem is the dimensionality of the problem is very high. So it's not an easy thing to do. But I think at least in principle, there's some very nice work that's been done. Heartflow Corporation, which does a lot of finite element modeling of tissue mechanics, has been able to substitute a lot of the detailed mechanical calculations with our AI surrogates. And there's a team at Yale trying to do diagnostics of aneurysms that's done some very nice work on AI surrogates. So those aren't quite the same. Of course, the neural network is more complicated than your ODE structure. But I think the idea of asking the question, can you reduce these complex models to simple underlying forms is very interesting. I know, what's his name? At Emory University has worked on this problem. Yanis Kevrakidis has worked on this problem, not using CompuCell though. I think it could be very interesting to do. I think it's a very profound question.
Oh, thank you. Yes, and yes, what this thing with space, like the Bronsolator and the whole idea about having different concentrations, different concentrations. What if it was not life, the species, chemical species would mix. So life appears to be somehow inherently spatial. Now, abstracting this away in an homogeneous model, it probably misses the very problem.
So what physiological models do is they don't represent space explicitly, but they do compartments. So you have a gut. A model of the gut would have many compartments, and then you pass material through that. And so effectively, you're creating a surrogate, a one-dimensional surrogate of the space with ODEs. And so the most sophisticated commercial pharmacokinetic models have many compartments. That when you swallow the pill, they'll model what happens in the front of the mouth, the back of the mouth, the throat, and each way down through the gut, down to the anus. They'll have each of those will be broken down into a block with its own pH, its own temperature, its own exchange with the environment. And so you can always, I mean, in a sense, since space is always discretized in a computer, in a sense, you're running finite difference versions of ODEs, which are simulating PDEs. So in a trivial way, we're always converting space into ODEs. The question is, are there simple ways of doing that? And I think that's the really interesting question. In the same way, trivially, a neural network model, neural network, cellular automata. Cellular automata are universal computers. So you can solve any problem with cellular automata. It's just not very efficient or convenient. In the same way, reaction diffusion equations are universal computers. You can solve any problem using reaction diffusion equations. It's not very convenient. And so the question, I think, is that makes what you're asking profound is, when is there a simple ODE representation of the result? The fact that there is, at least if it's not a stochastic problem, is always going to be there. It's always possible. So there's an existence theorem there. But actually making it useful is not so easy. And I think seeing when there's simple regularities and problems is really an interesting question.
Thank you. Thank you. And now a more technical problem, using CompuCell as a command line. Is it supported? Is it advisable? Is it?
Well, if TJ is here, so again, the CompuCell. So CompuCell has always had, not always, but almost from the beginning, had command line interfaces. And when we run large scale simulations on clusters, we run thousands of replicas remote. So so you can definitely call CompuCell with command line execution. Josh, maybe you can speak up on that.
Yeah. I mean, it's what you're saying is right. We can execute CompuCell from command line, and you can build sophisticated wrappers if you are familiar with it, so you can run parameter scans and so on using command line.
I mean, TJ and Josh have lots of Slurm scripts that will run thousands and thousands and thousands of jobs on a cluster. So
it works.
And TJ has been refactoring CompuCell also, so it's easier to call from Python. So traditionally, CompuCell has been good at calling other packages, but you've used CompuCell as the master package and then pulled the other packages inside of it. TJ has done a lot of refactoring, so you can do it both ways. So you can load CompuCell as components as a library in Python and then call it from other places.
Oh, that's great. I thought this was to be done some point in the future. It's already available.
I don't know if it's done. He's doing it, and you have to ask him how far along that project is. Maybe I'm old-fashioned because I grew up developing this code really as a desktop application in a time when everything was remote, and so I still tend to use it that way. But Josh and others will call it, and calling, for example, say a finite element package from CompuCell is possible. There's some nice solids modeling libraries or fluids modeling libraries, reaction diffusion solvers packages. If anything exists as a Python library, you can call it from CompuCell. Going the other way takes a little bit more work at the moment, but TJ has been working hard to make Python callable. And I'm pretty sure that most of it is now. Okay, we're actually almost out of time. I hope that those discussions, I understand that wasn't a perfect organization, but I hope that was useful. People had some good conversations. Was it helpful to have the time to talk a little bit? And if people were sitting in a room with nobody and waiting, I apologize. I figured if that happened for people for too long, they'd come back out and chat with the group. Let's see, Josh, we're supposed to end at 1:45, right?
Yes, and then next module start at 2:15.
At 2:15.
Right.
So does anybody else have anything else to talk about? As I say, I don't get to talk so much because usually I'm running the meeting. So it's a pleasure to get to chat a bit with people. You don't have to be asking me questions. You can ask your other participants questions. You can. Josh is here I don't know, Josh, if you wanted to join. I see Yajun has been sitting on his own. Do you want to talk to him for a minute?
Yeah, of course. Yep.
Let's see, where are you? Oh, you have to, have to move them myself.
Yeah, I can join. I can join.
Okay, great. Okay. Any other comments? I hope it's not, again, I know this is a little chaotic, but the hackathon organization tends to be chaotic. But a lot of times interesting things come out of them. Last year we had some, we've been working with a team from NIH, from NIAID, on a project that grew out of the hackathon last year, on Leishmania infection. If there aren't any other questions or comments, I guess we can say we'll take a lunch break. I'll leave the discussion rooms open for the next half hour or so, and then we'll reconvene at 2:15, and Josh will start showing you more details about how to use these subcellular models. Can I have a show of hands if you're on the call now? How many people are having this Microsoft the Windows problem with running Tellurium and Antimony? What fraction of people are having that problem? 1, 2. Okay, TJ said he thought he had a workaround. Did he talk to you about that? Okay, sure. And again, if I know it's not ideal, the two options that we have at the moment would be to roll back to the one version of CompuCell. So you could go to SourceForge and download the earlier version, which is not much fun, or to follow along today with NanoHub while TJ. I'm sure TJ can patch the source code between today and tomorrow, but I don't want people to miss the lectures this afternoon. So Joseph, please speak up and make sure everyone who's having a problem okay, Lorenzo, welcome back.
Hi.
Okay, I hope people had some productive discussions. Are there any last questions before we break for lunch? No. Josh, are you there?
Yeah, I'm here.
You want to queue up your screen share?
Sure, no problem.
And do you want, before we do that, do you want to take over as host? You're going to be giving all the afternoon presentations. So I'm not sure whether we need to have a well, I guess we should probably do our usual poll before we close, just to be consistent. Although I'm not exactly sure what the poll would represent for here. But Josh, I'll have to turn the meeting, make you the meeting host. Is that all right?
Yeah, that's fine.
Just remember that as meeting host, you can't join a breakout room.
Mm-hmm. Yep.
And you'll have to be responsible for the recordings and things.
I'm not exactly sure how to show the end of module poll questionnaire.
So you should be able to just launch the poll. It's a poll.
Okay.
So I'll leave this up for a second, but the main thing I'm concerned about is the recording. Okay, Josh, you're on the meeting now. You want to try out the poll? You should be, if you you should be able to click on poll and see the poll.
Yeah, I do see it, but I don't know how to
So
issue.
if you go to the bottom, you go to the bottom, it should say end. You can go to the top, you should go to the bottom, you should be able to see end poll.
Mm-hmm.
Okay. And now you go to polling in the control panel for Zoom, and you can do launch poll.
Oh, I see. And then choose
And there's
which
a
one I want to launch.
pull down. Yes, that's right. And so for your afternoon one, you need to use the number 3 and number 4. And once the poll is done, you have to download it and rename it.
Okay.
Okay. If we lose it, it's not the end of the world, but that's the only thing that's tricky. And I apologize to everybody listening in to have the chatter, but sometimes it's okay. Okay, good. I'm going to take a quick break, and I will be back in a few minutes.
Hello, everyone. We're supposed to start now, but we will give 2 or 3 more minutes for people to reconvene. Hello, everyone. I hope you all had time to catch something to eat or have a nice break. So I will be talking about, I will be presenting module 4.1. This was supposed to be the first module of today's state, but we ran over a little bit, but we will continue building up on what James was presenting. The module is going to be building upon what everyone, on what James was discussing earlier. But specifically in the context of how to model intracellular processes, although we will talk about how could this workflow could also be extended or used to represent processes that are happening at bigger than tissue scales, especially at systemic level processes. So again, this is the first module of day 4, and I'm suspecting this model might run over a little bit. I have a couple of interesting examples I would like us to walk through thoroughly. If we have a little bit of time after this presentation, then TJ will be presenting the principles of how to use Boolean networks in CompuCell. In the first part of the module, I will be talking about how we can use CompuCell to do multiscale models. And by multiscale models, we mean models that incorporate or integrate processes happening at widely different biological scales in terms of time and spatial scales. And we will learn how we can, once we've specified such models using antimony strings, how we can load these models into CompuCell, and then how we can load them into cells, depending on what the problem of interest is. Then I will be discussing 2 examples. I will so Tarun, no, because of 4.1.5 is just an example that we're gonna be using a repository on the internet, and that's me just walking you through how to look for those repositories. Then I'm gonna be discussing the delta notch and the cell cycle examples, and we'll go into the details of that later. So far we've focused on the capabilities of CompuCell of representing processes that are simulating processes that occur at the tissue level. So these mostly pertain the processes that involve the mechanical properties of cells, such as their where are they located, how much of the space they occupy, what shape they have, whether they move or not, whether they stick to each other, and whether and processes that we have roughly modeled just from this tissue level. We have modeled very basically, such as how cells can differentiate, how can they divide, how can they die, if they polarize or not. And we've also presented at the tissue level what the interaction between these specially resolved or specially distributed cells have with surrounding chemical fields in terms of how this might regulate cellular behavior. We saw that with controlling cell growth based on the amount of resources that the cell has available to them, and how the cells can secrete and uptake from their environment. However, the processes, biological processes, and the problems that us as biologists or physicists or computational biologists are interested often span over multiple scales, biological scales. Some of these processes, so again, so the reason the screen is small right now is because I'm planning on using tellurium I'm sorry, I'm planning on using computing NanoHub, so I have to have my screen split, and I was just showing this part. But again, the presentation is available in the Google Drive in case people want to have them as a reference side by side so they can look at the details more closely. But as I was mentioning, often the biological problems involve processes that span over different scales, processes that have occurred at the whole organism level, that interact with tissue level processes that in turn interact with processes happening at the intracellular scales or subcellular scales. And CompuCell has the capability of integrating these processes as we can model the processes and incorporate them into our simulations, depending, and how we incorporate them, it's gonna of course depend on the particularities of the problem of interest. And this in this particular image here, this was this is taken from a paper from our group. Sluka, it was the first author on this paper, and they were interested in looking at acetaminophen metabolism and the interactions between whole body PBPK models with models of the flow in the liver, and then interactions and the metabolism of the chemical compounds or the active compounds inside each cell. Julio, this is another example of a model developed by people in our group or related to our group. This one in particular was developed by Julio, and it's a multiscale model of somatogenesis. And it does also integrate multiple scales, specifically scales where something is being introduced into the cellular domain, a particular signal, and this signal then downstream regulates the differentiation and polarization of cells. When we think about the different types of subcellular modeling that could be of interest from the perspective or that could be integrated in CompuCell, we have many options. And in principle, we are able to integrate whatever model we want of subcellular processes with our CompuCell simulation. And the important thing, right, what is going to turn our model into a multiscale model is our ability to tie some of the properties of our CompuCell simulation to some of the outputs of the subcellular models. So for example, we could develop a model of a cell cycle where, let's say and we will see this later on, the state of a variable in that intracellular model determines or triggers cell division. And there is many ways in which we could implement this subcellular model. In this particular module, we will focus on modules to use on processes model using reaction kinetic models. These are the types of models that are antimony string support. But later on, TJ will be showing how other types of models, such as Boolean networks, could also be used to represent the same subcellular processes. Another interesting example that we will see later on is that delta notch. James already mentioned, talked a little bit about the notch signaling pathway. we will see how we can, using a simple set of differential equations to describe the state of the signals inside each cell, produce a model, a simulation that generates a characteristic patterning of delta node activation. So in terms of how to or where we integrate these reaction kinetic models, so there's multiple options available to us. And again, what options we decide or go with are going to depend on the particularities of the problem that we are interested in addressing. So for example, we can have a model, a subcellular model that describes processes that occurs in all of our cells. And in that case, we can have that model and we can load it in all of our cells, and then we can also particularize it if we want it. An example, for example, if we have a model in which all of our cells are capable of dividing, then we might incorporate a cell cycle model into all of our cells. If we have a model of viral replication and all of our cells are capable of taking up and reproducing virus, then we will load this viral replication model into all of our cells. We can also load the models by cell as a particular cell ID, let's say, because there is a special cell in our simulation that we want that cell to have a particular subcellular model. We could do that as well. We could we can incorporate it by cell type. So if there is just one type of cell that can that in which the process of interest occurs, we can just load it into the cell. So for example, we saw an example where we had a stem cell, and that was the only cell capable of differentiating. So a model of stem cell differentiation, we could load it into all of the stem cells in our simulation, and then all the terminally differentiated cells would not have that model. And finally, we can load the model as what we call a free-floating model. And in this is a special case. And we usually interpret these free-floating models as processes that affect the whole simulation domain more or less uniformly, and that describe processes happening at higher biological scales. So for example, if a compound is being we interpret something being introduced into our simulation as a compound being transported from the blood to the tissue, then we can simulate it that way. For example, if we had a model where the characteristics of our, let's say we have a infected tissue patch, this is something TJ described already, and we use information from that tissue patch to recruit immune cells in the lymph node, then we would use a free-floating model as a way to describing how information is transported or transmitted from the local site of infection to a whole organism level. There are multiple ways in which these reaction kinetic models can be specified and simulated, and how they can actually be integrated into compute. So we're going to focus on the Antimony specification that James discussed earlier, and we can then simulate these models in CompuCell directly. But there's other options as well. You can there is specialized software that can be used to develop such reaction kinetic models, and they could be translated into SPML files. And then these SPML files, which is just a markup language, use a standard markup language used to specify reaction kinetic models, can also be imported into CompuCell. And my last example does just that. So this is a lot in this slide, but I just wanted to give a quick overview, and we're going to go into the details of all of this. But the syntax and the reference to antimony models is always just going to look similar to this. We're going to define our antimony model as a string. Then we will import and load our antimony model. Either depending on the specific option that we're interested in, and we will explore some of those options. And then we always have to time step or call a special function to ensure that this that the CompuCell integrates our antimony model. So this time step underscore SBML is always gonna be it's gonna be required in all of our in all of the simulations that involve an antimony model or any type of SBML model. And there are ways for us, as I was mentioned earlier, the way we build this multiscale models is by connecting the outputs of subcellular models with properties of our CompuCell 3D simulation. And the way we do that is by retrieving or reading and writing the variables of these models. And then there is specific syntax that allows us to both read and write from the models that we have loaded into the cell. So the way I like to think about it is I imagine out my cell, and my cell has inside a model, and then I can read the outputs of that model inside my cell and do whatever I need to do with that read off at the tissue level. And then if there is a downstream effect from the tissue level into the intracellular processes, then I write on the same model output from the SBML model. And we will see, this is conceptual at this point. I just wanted to introduce the ideas, but we will see examples of all of these options in the next couple of exercises. So now let's go ahead and have our first exercise. So in this particular example, what we're gonna do is we're gonna just generate a genetic simulation template with two cell types, proliferating and quiescent. We're gonna specify the contact and volume constraints. I'm not sure we're gonna use the volume constraint. We're gonna specify both, but we're gonna, the contact and volume constraint. And then we're gonna define a basic antimony model. And then we're gonna load the model as free floating and make sure that CompuCell can step it. And then we're gonna retrieve the data from these free floating models such that we can plot them. And then, I'm sorry. After 1,000 Monte Carlo steps, then we're gonna change the value of manually. We're gonna change the value of our antimony model. I'm sorry, one second, I couldn't see my cursor. So the first thing is we're gonna generate the template simulation. We're gonna generate a new project. Again, this example is already loaded in the demos directory, but we're just gonna build it from scratch because we have time. So we're gonna generate our first exercise. No space. always forget to change the directory I can save stuff into. Okay. We're just gonna leave it like this because for the purpose of this exercise, we don't care that much about our simulation domain. We're just gonna add two cell types, proliferating and quiescent. We're not gonna have any chemical fields. And I mentioned that we want the contact constraint and the volume constraint. Yes, absolutely. So far I've just created a simulation. I didn't change any of the dimensions of any of the general properties of my simulation. I just created two cell types. Yeah, you can, I mean, the neighbor order, you can always, again, I keep forgetting that you can always prefer setting it to 3 just to avoid any square lattice effects. But really, for the purpose of this particular module, we're not going to be using the cells. We just wanted them to show that there's no interaction between cells and a model. And then we've selected contact and volume as the constraints we're going to use in our cells. Okay. Then I open my simulation, and then the next part I'm going to write a basic antimony model. And to write my antimony model, so I did wanted to say that this exercise will basically generate something similar to the template code that you were using in the previous exercise when you were writing your antimony model and plotting them to using CompuSol. So that's basically what we're going to build together in this exercise. So the first part, we're just going to define an antimony model. Let's call it model string. And it's always a multi-line string. And then I always like to type when I'm writing my equations, my parameters, and my initial conditions. So if I have to come back and read my code my model later, I know what part was intended to represent what. And this is a very simple model. In this model, A goes to B. We can think of a particle becoming B, and then a particle A becoming B, and then the particle B becoming A. So it's a bidirectional reaction. I always call them J's for the reaction rates. And that one is A goes to B at a rate that is K1 times A. And the next equation is, which is J1, is B goes to A at a rate that is, as James was explaining, mass action is proportional to B. For the parameters, now that I have defined these parameters here, K1 and K2, I have to specify what their values are. And I'm gonna set them to be 0.01 and K2 0.025. it has to be colon. Then my initial conditions, I'm gonna set them to 10 for A and no B. And I wanna make sure that everyone is with me here. Okay. So some people do need more time, so I'm just gonna wait a couple of minutes. But just as a recap, we had created a basic simulation where we've added two types of cells, so a proliferating and quiescent. We assigned them the contact and the volume constraint. We've defined a basic antimony model that describes a reaction where A goes to B at rate K1 times A, and B goes to 8 at rate K2 times B. Okay. So that describes the model for us, but this is just a string at the moment. It's not interacting with Compusol at all. It's just a string that we have specified in the step out. Now we need to be able to load it as a free-floating antimony model. And again, when we talked about a free-floating antimony model, it's a model that is not attached to any object of our simulation. It's free float, and that's why it's free floating. We can still, Compusol is still gonna step the model, and we can still interact with the model, but the model is not attached to any of the objects of our simulation. And if we want to load that model, we always do them in the start function. And to have access to the code that would allow us to load the model, we always come to your friend CC3D Python, and we go to SVML solvers. It's a little bit of a long list here, but the ones we are interested in is this add free floating antimony model. There's many options depending on the type of markup language or model specification that you're using to specify your subcellular model. But in this part, as I mentioned, in this particular exercise, we're gonna focus on antimony models. So we add this free floating antimony model, and then it has a, let me decrease it a little bit. So it has a little bit of information for us. It says that if we have a file, we should name that file model file and have the path to that file. So if we have the antimony string in we haven't specified it up here in our same step out, we could have it as a independent file, save it in an independent file, and we could load it that well, referencing that file, or we can reference directly the model string. And it tells you what the structure of that basic structure of that model string should be. So if we look at the options that add free floating antimony model has, as I mentioned, there is a model file. If we have a path to a file that contains our antimony model, but we do not because we wrote our own model, our model string. We already called the model string, so this serves as the input to as this input to the function. then we have to come for a model name. And this part is important because the model name is what we're gonna use to reference the model later when we want to retrieve, write and read the data or values of our, or any parameter values or variable value, state variable from our model. So for this particular example, I called it my model. Step size is another important concept, and it refers to how many the basic time step in CompuCell, as it was discussed some time ago, it's called the Monte Carlo step, or what we call the MCS. Now we have to specify, and this part is a little bit too complicated, but for now we're just gonna set it. We're just gonna describe it shallowly by saying that the time step specifies how many of the internal units of the model corresponds to one Monte Carlo step. For now, we're just gonna set that to 1. But this becomes very important. So 1 unit in Monte Carlo step corresponds to 1 unit in the intracellular model. So let's say my Monte Carlo, I said at I arbitrarily decided that a Monte Carlo step is equivalent to 5 seconds. Then I'm saying that each unit, each single unit in the subcellular model is also corresponds to 6 seconds. But this becomes very important when you want to not just integrate but also synchronize the units of your tissue level model with the units of your subcellular process. For initial conditions, we're not going to specify the initial conditions because we have already specified them in the Antimony string. And then that way, we have already loaded our Antimony model. So if we run this, should be able to run, although I'm not CompuCell is solving the model, but I'm not doing anything with it. So I'm not gonna be able to see anything really, just my cells. So I can see my cells. Right, but there's really nothing interacting with them. And I haven't, although CompuCell is actually solving the SBML model, I have no way to see how that is sort of like hidden at the moment. So what we will do is to be able to see how CompuCell is solving the SBML model is that we're gonna plot the values of the state variables of my model. So that's the next step. Oh, I forgot. Actually, I made a mistake. A mistake that I did. I mentioned that it was very important, and I absolutely forgot. So, any ideas of if I ask the question now, is CompuCell 3D step in the model or not? What is the answer? No, it's not, because I forgot to actually call that important function that it's supposed that I am always supposed to call when I'm using SBML models to step the model. And TJ, I'm sorry, Tarim has an important question. I will let TJ answer that question, but I'll just continue with this while TJ can explain that in the chat. So I forgot to time step my model. So if I want to get the code, I can type it because I know it already. It's not that complicated. But if I forget it, I can always come to CC3D Python, SBML solver, and then there is this time step SBML models that allows me to time step the model. And now CompuSol is time stepping the model. And if I rerun CompuSol, it's just gonna be the same because I'm not interacting the model. But in this case, now CompuSol is actually time stepping the model. And I'm gonna relaunch the poll just to make sure that everyone is with me up to here. Okay. So yeah, most people seem to be with me here. I'm just gonna continue on. The next step is that we actually want to be able to see how CompuSol is stepping the model. And to do that, what I will do is we will create plots and we will retrieve data from this free-floating model, and then we will add it to our plots. And this should be, by now we have done this a couple of times, so I'll go fast over it. For scientific plots, we always come to CC3D Python, scientific plots, setup. I called it free floating model variables. I turn on the grid just as an exercise to show that you have some ability to modify the options here. And then I created two data series because I have two variables. One is A, the other one is B. And then I want them both to be dots. And then red and green is fine, or red and blue. I call them different. I had a different setting. So that it's adding the plot window. So remember CC3D Python, scientific plots, and then I just rename the title of my plot, and then I created two data series, A and B, such that I can add the data from the variables A and B. I run the model now. I should get a window, an additional window that it's my plotting window. But if I run the model, I shouldn't see anything, right? Because even though I created the window, I haven't passed on any data. To pass on any data, I have to do that in the step function. So now, and this part is important, I'm gonna add the data points to the plots. And to be able to do that, I need to be able to retrieve or reference the model that I've just loaded. So I'm gonna create two variables, A and B. And on A, I'm gonna read the value of the variable A in my model. So the way I do that, I reference that is by this self SBML, my model, and then the name of the variable. That I type directly because it's very easy. But again, if you do not know what the reference is, again, you can always come to SBML model, and then you get the SBML value for a free floating model. And that is supposed to give you the same syntax. When I use it here, and see that now where it says model name, I have to change it to the name of the model that I specified when I load it, and then the variable that I am interested in reading. And now that I have, now every time step, I'll read the value of the variable A in this free floating model. The next step is adding these variables to my plots. And I always come to scientific plots, add data points. These ones, since are a little bit long, I do not type themselves manually all the time. But I know that now the data, I have two data series, A and B. And then I store the values of the variables in the free floating model in these variables A and B. And now if I save and relaunch my simulation, I should be able to see the so Farnoosh, I did it really quick, but when you loaded the model, there is a variable called initial conditions that I deleted because I have already defined the initial conditions in the Antimony model. So you can delete that part of the function or the class instantiation. So yeah, so now I'm able to plot the model. And the reason I included the cells here is because I wanted to show that the cells are in this free floating model. I created the free floating model, but I really haven't done anything with my cells. So the model is basically being stepped and resolved by CompuCell, but it's not interacting with myself in any way. So the simulation shouldn't be affected by what is happening in this free floating model or my CompuCell simulation. Okay, I'm gonna relaunch the poll just to make sure that everyone is with me. So so is there any, Polly, there seems to be, maybe there is a problem with your model specification in XML. So I'm gonna, if you need, is there anyone available to help in a breakout room? In the meantime, I'm waiting on other people to catch up. Okay, so this is basically the same code you had in your previous it is not in the medium, right? It is global in the sense that it's not in all cells. It's just independent of every object in our simulation. So basically, it's running parallel to my simulation, but it has no interaction whatsoever with my simulation unless I specify it. But it's not in the medium. It's not in any of my cells. It's not on all of my cells. It's basically nowhere. And that's why it's useful to model processes at larger than the tissue scale, because this will happen basically, let's say above my or in the scales up or above my simulation. So that's why that free floating is a good term to think about it. So I'm sorry, because I'm also assigning to breakout rooms. So okay. Okay, well, so as I was mentioning, this is basically the same simulation we were playing with in the previous module. It just had the option for you to specify your antimony model, and then it was plotting the state variables of your of the model specifying the as a free floating model. Let's do something a little bit more fun. I'm sorry, I do not see. Right. So now we can show, now that I've read from my free antimony model, I can actually also write to my free floating antimony model. And the syntax will be basically the same. So let's say that at time MCS 400, I want, for whatever reason, my variable A, or the value of, I'm sorry. I'm using a Mac on Nano Hub, and then I forget that they're not the same keys. So let's say that at time 400, we want our model, we want the value of our free float, the value of A in our free floating model to change. So what we do is that we use the same reference, but now we set the value instead of reading it. Okay. And then now I can save it's as easy as that. And then if I rerun my simulation, I can see that it's CompuSol is stepping the model, but when it reaches 400, then the value of A changes, and consequently the value of B will change as well, of course, because there is an interaction between A and B. So the equilibrium concentration of the two variables changed. So for those of you who need this as a reference, this exercise have been uploaded as exercise 411 in the Google Drive. Okay, I'm gonna give a couple of minutes for the people who went to the breakout room to come back, and for people who are still working on the exercise to catch up before we move on to the next exercise. In the meantime, if anyone has any questions, please feel free to ask.
can you show your code again?
Yes, 1 second. So Ben, I'm actually not exactly sure. I think that's a little bit of leftover from previous version, but I think the state, you can reference the state of the model.
So I think state is just referencing the whole model.
Oh yeah, yeah.
With
So it's how to reference the
whatever-
model, right?
Yeah.
Because if you look at the syntax that it provides, it's exactly the same syntax that we use to reference the model plus the variable of interest. So it's basically returning the model or the state of the model as an object. Technical terms. Okay, I'm gonna continue with the next couple of exercises. In the next, managing the Zoom meeting while presenting, it's actually more complicated than it seems. I have like 5 different windows all on top of each other. So in the next exercise, in the previous exercise, we loaded the anti-money model as a free-floating model, right? And I explained a couple of times that there is particular uses for that type of model integration. But if we want to model subcellular processes, the more natural way of coupling our CompuCell simulation with our models is to load this subcellular models into each of our cells. So this is what we will do in our next exercise. We will just continue building up on this same code. Yeah, sure, let's do it. Let's continue building up on this code. We're just gonna have windows that we're not gonna be using. The syntax to let's stick with the same anti-money model, but the anti-money model now we're gonna interpret as something that is happening in a reaction that is happening inside cells. So actually, please follow me what I'm doing next, because we're not gonna be able to use the plotting. Please delete the syntax that we use to create the plotting windows, and the syntax that we use to add data points and to reference the values of the SBML model, of the free floating model. So basically, we're just gonna leave the time step function inside the step function. And then let's go ahead and delete everything from the start function as well. So basically, to start this example, let's just start with a blanket start function and then a step function that just has this time step, the time stepping of the SBML models. So let me just make sure that everyone's with me there. So I didn't do anything fancy. I just deleted everything from my start function and basically everything from my step function. So I can use this as a blanket simulation instead of having to create a new one. As I was mentioning, we have the option of loading this antimony model that we have specified inside each cell or inside a cell, depending on if we interpret the model as a reaction happening inside a cell, whether it be a signaling process or a gene regulatory process or even a metabolic process. So when we interpret them that way, then we can the most natural way of integrating CompuCell with those models is to load them into the cells. And to load the model that we have already specified into the cell, we just come back to CC3D Python again, SBML solvers, and then we have three different options here. We can add the antimony model by cell ID. We can add it to by cell type, or we can add it to individual cells. And we will start with adding it to a cell ID, to a model. We will start with adding the model by cell ID. Again, I get the code snippet and it looks very similar. It's just changed. Instead of add free floating antimony model, it now has add antimony models to cell ID. The model file, like explained already, we wrote it, or we wrote the model string, so we don't have to use that one. The name of the model, let's again call it my model. And let's just model to cells that have ID 1, 3, and 7. We have more than 7 cells, so we won't have a problem. But this is basically, the cell ID is the identifier of each of the cells in our simulation. It's a number that identifies each of the cells. It's basically the identity number. So in this particular case, I'm gonna upload this model into cells 1, 3, and 7. Step size, let's again make our step size being 1. And again, initial conditions I specified in my antimony model, so we can get rid of that part. So all I need is the model string, the model name, and the cell IDs and the step size. So good question. You see that this is a this is a list, right? The data structure is a list. So cell IDs is expecting a list as an input. I could pass a list in that particular case, Tarun, that goes from 0 to, let's say, 100 or 1 to 100. And that would generalize it to all of the cells with ID from 1 to 100. But we're just gonna stick to 1, 3, and 7. Okay. So what we will do next, I did all of the options at the same time, but we're not gonna do that now. What we'll do is that we are gonna, we want to be able to visualize the state of the subcellular model, in all of our cells. There's multiple ways we can do that. We could potentially plot the value for each of our cells, but then if we have many cells, that would be a lot of plots on top of each other. That might not be meaningful. We could plot the average of the values. That's fine as well. But in this particular case, because this stage is pretty exploratory, what I'll do is I'll just plot them visually, so I can see, I can color my cell depending on the value of the particular variable of interest. So to do that, I have to create a auxiliary field. We did this already yesterday, but we're gonna review it. To create, and remember that again, the auxiliary field, what it does is basically creates a window in our simulation in player where I can display the value of a variable inside a cell depending on the value specified in the dictionary of that cell. So to create this auxiliary field, the first I always do it in the init function. I come to CC3D Python, extra fields, automatic tracking, track cell attribute. And we're gonna track two. We're gonna track the value of A in our cells and the value of B in our cells. So we need two of these fields. And let's call it the field name. Let's call it A and B. And the attribute name is a reference to, yes, it's CC3D Python extra field automatic tracking track scalar cell attribute. And this has to be done in the init function of the step hour. And then the attribute name has to reference the attribute in the cell dictionary that I'm interested in displaying. We haven't created them yet, but we will call them A and B. So let's just call the attribute name A and B. So the field name is A and B. The attribute names is also A and B. But now I have to create and update this attribute in the cell dictionary. And that is just as simple as in the step function, adding the attributes, I'm sorry. A and B to my cell dictionary. And then question for you, how do I reference the values of the subcellular model yes, I'm sorry. Yeah, I have, right, cell is a reference, so it needs to be let me see. Right. I'm sorry, I created, okay, yeah, this is just a little bit more complicated that I wanted to do it, but I'll just do it in the step function, and I'll set them to 0 for now, and then I'll explain how do I reference them later. So let's, and I'm sorry for the confusion here, but what I'm basically doing is each step I am iterating over my cell list, and I am updating the value of this cell dictionary attribute called A and B, and I'm setting it to 0. If I come back and I run the simulation now, now I can see that I have the fields A and B. If I run the simulation, there's nothing is going to happen because I haven't tied up the intracellular models to the values of these cell dictionary attributes. But I've only created the cell attributes. I'm gonna make sure that everyone's with me. I'm gonna put the code back. So the people who need help, feel free to ask questions here, or I can assign you to a breakout room if preferred. Okay. Is anyone available to help Tarim? Okay, I'm gonna assign to your breakout room. Okay, good. So, and this is why I wanted to give people time to catch up, because this part is a little bit complicated and I do not want to get into the details here, but the basic idea is that we have just loaded the antimony model into some of our cells, not all of our cells. We have loaded into cells number 1, 3, and 7. If we try to reference the antimony model on a cell that does not have one, we're just gonna get an error. So the way my way around it was to use this this exception handling, and I'm not gonna go into the details. So we actually good question, Polly. Good practice is always to initialize the variables that you're gonna be using in your simulation. Excellent point. So, right. But really, if you think about what the flow of the code is, this step function is called when Monte Carlo goes from 0 to 1, and if the dictionary did not have a attribute called A, it will create it at that time, and that's why I didn't create it in the start function. But yes, it's always good practice to create the variable, initialize it in your start function. Okay, so back to this. So again, what I was trying to explain is that we haven't loaded the antimony model into all of our cells, and if we were to try to if we were going to try to reference the antimony model in a cell that does not have it, we will our CompuCell will issue an error. So I did some exception handling here. I'm not gonna go into the details. This is a little bit more Python-y than CompuCell. But what I'm basically saying is here, if the cell does not has a antimony model, just keep going, do nothing. But if the cell has an antimony model, then I want to update the value of the dictionary with, right, we could also do that, but I generalized it because of the other exercises that we're gonna have later. But yes, you could actually iterate through cells with ID 3, 1, 3, and 5. There's many ways you could handle this this complication. But now the question I wanted to ask you is, now that I have handled that problem, if I want to update the value of the dictionary attribute A, how do I reference the model inside that cell? I mean, the answer is there, but I also wanted you to give it a try by looking at the CC3D Python. So we're gonna reference the model inside each cell by typing cell.sbml, the number of the model. That's not the format. Note, in the previous example, when we loaded the model as a free antimony string, do you guys remember what the reference was? If not, can someone pull it from their CC3D Python pull down menu and type it in the chat? What was the reference for the free floating antimony model? Right, it was self.sbml model. That references any free floating antimony model. If we want to reference the model inside each cell, then we had to get the reference to the cell and then reference the model inside that cell. So now we can reference the values of the variables inside the cell. And then when we come to CompuCell and execute the simulation now, we can actually see what the value of the variables A and B are inside each of the cells that I initialized the SBML model. The reason they look both red, again, it's because my visualization options. So I can come back to my visualization properties, and I know that A and B can only go to 10. 10 is the maximum value they can go to. So I can readjust the color bars. So I can see the relative values of each variable in the cells that are initialized the SBML model. And I'm gonna relaunch a poll. just to make sure that everyone is with me. Good. So this exercise is available to you as exercise 412 in the demos. The same way we did with the, so we loaded the, model. So we've loaded the SBML model as by cell ID. In the example that I have all of the examples of what it looks like when you load them by different options. We'll try some of them to see how the syntax differs from the one that we've used so far. So another option that we have is to load the Antimony model by cell type instead of cell ID. And to do so, we come to CC3D Python SBML solvers, add Antimony model to cell types. And again, I have to fill up what the function, I have to, the function inputs, I need to fit them correctly. Our model string, our model name, we're gonna rename that the same way so that we can reuse the code. And then now I have to specify instead what the cell types that I wanna load this model into are. And you might recall that we created two different cell types. We call them proliferating and quiescent. Let's just load our model in our proliferating cells. And we have to type the cell type all in caps. And again, we have to specify what the step size is, and let's call it 1, and let's make it 1, and initial conditions we can get rid of because we are specifying them in the Antimony string. Now, if I come back to the simulation and I reload the simulation, so I can see that blue are proliferating, green are quiescent. And if I look at the value of A, it's only loaded in the proliferating cell type and not in the quiescent cell type. Relaunch the model and show the code. Some people, there seems to be a decent amount of people that need time to work on this. So I'm gonna just give a couple of minutes to people to catch up. Again, I want to remember everyone that this exercise are available for you in the Google Drive, and it's called exercise 4112. And while we wait for everyone to catch up, I will be happy to answer any questions. I'm not sure, Ben, I'm not sure there is a satisfactory answer to that question, except that maybe there is a particular reason why Juliano might know, but I actually might think that it's the way it was done at the beginning and it's the way it's been done since. Right. The end of line 33, 36, model string, the model name, mile name, and then the cell types that I want to attach those SBML models to. Right, Joseph, if you, right, because I fixed the values of my color scheme, they look teal and cyan because I fixed the values to be 10 and the actual numbers are lower, which is like equilibrium numbers that we saw earlier. If it's automatic scaling, always the highest value is gonna be red and then the smallest value is gonna be blue. So they will both look the same. The reason I fixed the scales is because I wanted you to be able to relate the previous values in the equilibrium concentration where A was lower than B to the colors that were shown in the visualization windows. Okay, so so I do want everyone to catch up with me because it gets more complicated from here on. So I'm just gonna relaunch the poll to make sure that at least most people are with me, or at least are just watching the presentation before moving on. Also, you can click yes if you think it's time for me to move on to the next exercise. Okay, so there is, as I mentioned earlier, there is many other options that we can use to load the antimony models. I'm not gonna do them all now because it's a little bit repetitive. There's more interesting examples to work with, but I'm just gonna let you play with it as a homework. You can try loading the antimony model to all of my cells. And for that, what I do is that I loop over my cell list, and then I use this syntax called add antimony model to cell. And then when it requires me to load the cell, I load it to the cell that I'm referencing. There is a typo here, actually. It shouldn't be cell 20 that's just because I copied from the previous line. It should be cell, which is the reference that I'm using to the name I'm using to reference each of the cells in the cell list. And that will do is that will load the antimony model into all of my cells. So there's a little bit of a subtlety there, but the idea is that if I want to load it into all of my cells, because it's a process that I think happens in all of my cells, all I need to do is to loop over my cells and then load the model in each of the cells. Okay, so now let's move on to a little bit more biologically relevant exercises or examples. In the next session, in the next part of this module, we're gonna see how we can use the principles discussed up to now to model delta notch, and whether we can reproduce the patterning characteristic of the delta notch signaling. And we will show how we can actually add steering panels that allow us to change the parameters of our SBML models on the fly. And again, James was talking earlier about how CompuCell can be used in two rather distinct ways. One way of using CompuCell is, which we tend to use at the very beginning of when we're building models, is exploratory. And in that, when we're using CompuCell in an exploratory mode, we tend to rely a lot on the visualization, on how the simulation looks like. And then steering the parameters on the fly allow us to identify what parameter values better match the expectations that we have in terms of what the simulation should look like. Once we've settled with the parameters and the mechanisms that we've incorporated, we move on from there and we rely much less on the actual visualization. But for the exploratory model, we do rely a lot on this point, on the visualization and the steering. I'm gonna go very quick over delta notch. I'm not an expert on delta notch, but just as a quick introduction, delta notch is one of the most important cell mechanisms for cell signaling. And notch specifically is a transmembrane receptor that coordinates and activates the signaling through the notch pathway. And the notch signaling pathway is associated with different downstream effects in cells, including the regulation of cell fate and formation at a tissue level of pattern. If you want to have a better idea or more, get a better review of the delta notch signaling pathway, there is a nice reference here. In terms of molecularly, what's happening in the delta notch, the delta notch is that it's acting as a molecular switch. Both delta and notch are transmembrane proteins. Delta is the ligand of notch. And then delta works as a signal from the signaling molecule onto the signal receiving cell that has a notch, that or where notch is activated. Sort of trying to keep track with the chat. So, and then once notch is activated in the receiving cell, then this activation leads downstream to the inhibition of delta in that same signal receiving cell. So basically, delta from a neighboring cells activate notch in the delta in the receiving cell, and then the downstream effect of the activation of notch is it inhibition or downregulation of delta on that same cell receiving cell, signal receiving cell. And that leads to this usual pattern formation where we see high notch, high delta, high notch, high delta, high notch, high delta, or high notch, low notch, high notch, notch, low notch. There's been a couple of mathematical models explained, proposed to model this delta notch activation. This is we're gonna for this example we're gonna use this model published by Collier, and it's a very simple model. It just have two differential equations, one describing the activity of delta and one describing the activity on notch in a single cell. I wanted to quickly explain how to interpret these equations. It's a little bit beyond the scope of the class, but just as a quick readout, once I see these equations here, the functional form of these equations, I immediately interpret them as activation or inhibition. And that is what this is doing. So we can read this as delta activating notch, or this delta bar activating notch, and then notch inhibiting delta. And delta bar is a parameter, it's not a variable, it's a parameter of the model, and represents the average delta signal that a cell receives from its neighbor. So remember that this two equations, sorry about the exterior noise. These two equations are describing, and this is important to remember, the state of the delta notch signaling inside a particular cell. In a particular cell, how much notch activated there is depends on how much delta is in their neighbors, which works basically as an input parameter in my in the delta notch model, and then the state of the notch variable determines the amount of inhibition on the delta variable. Okay, and that's all, and with that model we can actually reproduce the special patterning of activation inhibition that we see in the delta notch signaling. So what we'll do for this example is we will create a very small simulation domain, just one single type of cell, and we're gonna assign them both volume and contact constraint, and we're gonna fill the whole simulation domain with our cells, so there would be no open space. And then we're gonna write an antimony model that corresponds to the ODE equations that we just saw. James discussed a little bit of that earlier. We're gonna put that in practice now. How do you go from ODE equations to the antimony string? And then we're gonna load the antimony string into all the cells and initialize the value of delta and notch. And then we're gonna calculate what this delta bar or D bar represents for each individual cell. And then we will again create auxiliary fields that are gonna help us see the level of delta and notch in all of the cells that we created. Okay, so we're gonna So we're gonna build the simulation together from scratch. So CC3D project, new project, let's call it Delta Notch. And I said it's gonna have 42 dimensions. And actually we want our boundary conditions to be periodic on X and Y. Our membrane fluctuations of our pixel copy range, not our membrane fluctuations, to be range 3. And we want the uniform initializer, which is a rectangular slab. We're just gonna create one type of cell. Let's call it my cell. No chemical fields. And I said that we will have contact and we will have volume local flex, so we can specify the volume constraint parameters in Python. And we're gonna finish the simulation. So let's make sure that everyone has a blanket simulation to work from. The plugins are volume, local flex, and contact. Okay, I think everyone is with me now. So let's close this exercise 1. Let's open Delta Notch. And so in the XML, we should have the dimensions of our simulation, the boundary conditions on X and Y, our 2 cell types, our cell type. And then we want to create, we want to fill the whole simulation domain. So we're gonna go from 0 to 42 and 0, I'm sorry, I always type them incorrectly. 0 from 42, 0 to 42 on X and Y. So we can fill the whole simulation domain. I didn't do anything here. Let's just launch it to make sure that that we have a working simulation, and that it's filling up the whole simulation domain. Yep, it is. Okay. So let's make sure that everyone is with me here. Yeah. Okay. So yeah, pretty much everyone is with me now. So in the next step, we're gonna have to turn our antimony model with our antimony model, our ODE into an antimony model. And I'm gonna ask this question so so you can answer in the chat. How many of you want to have time to work that out yourself, as opposed to me doing it for you? So I was thinking on giving you time to. It's not complicated, but I wasn't sure if you prefer me to do it right away or to give you time to try to figure it out yourselves. Okay. So let's do it together then. So, and this builds up on what James was explaining earlier. So there is a way we can, there is a simple way to return, well, I mean, knowing the syntax of the antimony string would allow you to turn this ODE equations into an antimony model. The first thing is we want to define the model string. We haven't defined them yet, I'm sorry. And that is a good point because we were supposed to define them in the start function. So in the start function, yeah, we said that we will, as for the target volume, we'll set it to 50. And then the lambda volume we'll set to thank you, Polly, for reminding me. So now we can come back to writing the model. And I'm not gonna spend a lot of time into doing it, but again, I always have the, it's always good to comment on your string so you know which part corresponds to what. Divide it into your equations, your parameters, and your initial conditions. Now, in these equations, notch is not becoming delta. Notch is regulating delta. And the way we end, same with D bar is not becoming notch. D bar is up-regulating notch. So the way we write those types of regulatory interactions is by, we call it nothing going to the variable. So it's gonna be n and going to be d. So this is basically the up-regulation or inhibition of notch and delta. We call it also nothing goes to n and nothing goes to d. And the regulatory parameter for notch is gonna be D bar over D bar plus A, and the inhibitory interaction in delta is V times 1 over 1 plus beta times n to the h, right? So the regulatory interaction on notch is d bar over d bar plus a, and then the inhibitory interaction of notch on delta is b times 1 over 1 plus b plus n to the h. Let's make sure that everyone is with me here.
There is minus d and minus n there in the equations, but
Yes, we will type them now because we were first typing the first terms, and those we could directly type them as minus d and minus n, but I also wanted to show that since they represent decay, we could also type them differently. It's equivalent. So let's go ahead and do it now, and then I'll just explain what the equivalence is. So in n, we have a decay term that is called n, and I can just type minus n. And then in d, there is a decay term that it's d times v, right? So it's inside the parentheses. But yeah, as I could have typed them as well this way. And those two, this and this, are equivalent to this part. So they are doing exactly the same function. Just gonna delete them from there though. Or I can leave them and comment them out, but I'm just gonna delete it to avoid confusions. Okay, now I have to type the parameters. A. It's gonna
if
be
the equation
0.
is kind of second order, can we write in this antimony in this part? If it is the second order, the equation, is it possible to write?
I mean, if it's a second order, like in n, like
D,
like in
d2d over
right.
d2.
Right. Yes. Yeah. The specific, like if this is, for example, the easiest way of just taking the ODEs and writing them up here. So you could potentially do it as well. Like let's say it could be d square here, or n square here.
I mean, in the derivative term in the first part.
Here.
And the equation in the left part. Like the second order derivative, I mean.
I'm sorry, I did forget this part. Is this what you're talking about? Because I did forget to write this. So it's d bar to the k, and then n to the h. So, but yeah, I'm sorry, go ahead and ask your question again.
I'm just talking about the second order ODEs to model it like that. Is it possible?
So so can you type an example in the chat of what the equation will look like? What type of equation do you have in mind? And yes, for people who are asking, I did forget to type the d bar to the k. So yeah, I do want to answer your question. I just didn't understand what the, yeah. Oh, if it was, okay. Right, I'm not exactly sure. That's an excellent question. I think there's people in the group in the seminar that are more right. Now I do understand. My guess is gonna be yes, but I'm not exactly sure because I already have a reference to the rate, right? So I could potentially reference the rate in my equations, but I'm actually not sure. That's an excellent question. So if I make it as a separate reaction. But again, yes, I mean, I can I would have to, I could. If I type it this way, right? The rate here, I can type whatever I want. I can make it d squared, or I can reference, let's say, I'm guessing, but I've never tried it, that I can potentially reference the rate previously defined or any other functional form that I want. I can define a function and then use it here. So I'm free to specify the rate at whatever rate I want. But I'm not necessarily sure if I can actually reference the rates themselves for, let's say, second derivatives or second order derivatives. But there's people who are more of experts in tellurium in the chat that might actually be able to help with that part. I'm actually not sure if I can make my functions, my rates, depending on other rates, for example. Yes, there is a way of changing the values of the model parameters, just the same way we have referenced the values of the variables and we have set them. You use exactly the same syntax, but instead of referencing the values themselves, you then reference the parameters. So let's just continue here. Again, questions that I might not be able to answer directly. There's James is coming. He'll probably be able to answer, and there's also other people in the group that knows in the chat that know better. So let's continue specifying the parameters of the delta notch model. And then for the initial conditions, I'm just gonna set them to 0 here because I'm gonna initialize them elsewhere. Okay, so this would be the antimony equivalent in, I forgot K. K equals H, right? K equals 2. There you go. So this would be the equivalent antimony string of the ODE equations displayed here. I call the average in the example, but it's the same as the bar. Yeah. So let's call D bar. That's another good question. D bar, let's set it to 0.4 into a 0.1. Right. Thank you everyone for the reminders. I think I'm just gonna need a break here soon. So making a lot of mistakes. So but let's just try to get as far as we can in implementing the delta notch. So yeah, this is the equivalent. D bar is a parameter in this model, right? Because how do we know that D bar is a parameter? Because there is no rate equation that describes the change of D bar. So that it's interpreted by the Antimony parser as D bar being a parameter. Okay, and then in the next part, we're just gonna load our Antimony model into all of our cells. And we do that in our start function. Again, CC3D Python, SBML solver, add Antimony cell, let's say to cell types. Let's use the cell type syntax. And we're gonna load it into we're gonna again fill up the inputs on that function. I actually do not remember how I called the model string. I called it model. Let's call it model string. And let's call the model name is gonna be DN for delta notch. And then the cell types that I wanna load it into, if we remember, the cell type we created is called my cell. And then for the step size, we will use here 0.2. And why 0.2? It's because I wanted to slow down the frequency at which the signals was being updated in the cells. But that's just, I mean, that's a little bit more advanced. It doesn't matter for the purpose of the module what the step size is. So it's a matter of how much resolution do you want. And again, we always want the integrate function or the step function inside the step in our step our CC3D Python SBML solver time step SBML model. So that will load the SBML model into our cells, and then yes, Polly, we will later update dbar reading information from the simulation. But for now, we just have to initialize it with some value. So we just put a value there, but we will update that value by reading the delta in our cells later on. And that's how we're gonna turn the delta notch model into a multiscale model. But let's just, so this part finishes the loading. Yes, I did. No, I'm sorry. No, I did not. It's outside because I'm loading it by cell type. So I don't want to load it. I just need to load it one time. I don't need to load it every time I iterate over my cells. To call time step SBML, CC3D Python SBML solvers, time-step SBML model. Okay, and what we'll do now is let's take a 5 to 10 minute break and let's come back and finish implementing the antimony model. Gonna launch the poll just to make sure that everyone is catching up. Okay, we'll give everyone 2 or 3 more minutes to catch up, and we'll continue with implementing Delta Notch. Okay, well, welcome back, everyone. Let's continue with our previous exercise where we were building a multi-scale model of delta notch signaling. So far, we've defined our delta notch model from the ODE equations. We have specified what the parameters of that model are and what the initial conditions. And we've loaded our antimony model in all of our cells. In the next exercise, as some people pointed out already, now we have to tie one of the parameters of our model, the delta bar, the D bar parameter, to some of the properties of our CompuCell simulation. And in particular, the way we're gonna tie this D bar to the CompuCell simulation is that we will, if we have a single cell, we will look at all of the neighbors of that cell. We're gonna see what the delta value is in those neighbors. And then we're gonna average over the neighbors of the cell. And that's gonna be the value that we pass to D bar. Again, just to make sure, because this part is really important, the way we're gonna calculate D bar inside each cell is that we're gonna look at each of the neighbors of the cell, see what the delta value is in each of those neighbors, and then we're gonna average that delta value over the neighbors. So we're gonna have an average delta from their neighbors and pass it on as a D bar to that cell's delta notch model. Does that make sense? Want to make sure that makes sense because it's really important. It's subtle, but it's actually the way in which we are building and the multi-scale aspect of the model or of this simulation. Anyone need me to explain it again?
I would appreciate you explain again. Thank you.
Okay, let me see if I can draw Because that's probably better. Yeah, I do. So we have a cell, right? And then inside that cell, we have a delta notch model, right? So we have a value for D and we have a value for N in that cell, inside that cell. And that cell has multiple neighbors that also have a DN model inside. And all of the model have the same structure in the sense that they all have the same equations and the same parameters. One of the parameters in this DN model is the delta bar or D bar, the D bar parameter. And the way I'm gonna connect or make this cell multiscale or this simulation multiscale is that the delta bar in this parameter, in this cell, I'm gonna calculate it by looking what the delta value is in each of their its neighbors. And then the delta bar is gonna be the average delta of the neighbors, average delta. That's what the D bar is, the average in the neighbors. Does that make sense? So each of my cells has a single instance of the delta notch model, which means that each of them has a D and an N, a delta and a notch. And then the way I calculate the parameter D bar for each of those cells is that I look at their neighbors and average the delta of their neighbors. Okay. So the way I do that is, as is explained in this code, is that I'm gonna, in the step function, I'm gonna iterate over every single one of my cells. Then I'm gonna create a variable that I'm gonna call the average, set it to 0, and a counter, and set it to 0. And then now that I'm at a particular cell, I have to visit or get the reference of each of the neighbors of my cell. And if I want to do that, there is a convenient Python script that allows me to do it. I go to CC3D Python, visit cell neighbors. Okay. And then this for loops is looping over this get cell neighbor data list, which is basically the list of the neighbors of a cell. And if the cell has a neighbor, right, then I will do something. And what I'll do is that I will add the delta value in that neighbor to my the average value variable. And the way I reference, right, I define a D bar inside the SVML model, but that's independent. I will have to, and later on, I will make the equivalence between D bar in the SVML model and the average here in the Python code. But I named it differently at this example just to make it clear that they are different. They have different functions. So the average what I need to do now is I need to get the value of the delta in the neighbors. And neighbor here is basically has the same function as cell in this loop, except that it references, instead of referencing the cell, it references the neighbors of a cell. So I can use this neighbor reference the same way I use my cell reference in the loop to read the value of the delta in the cell, in the neighbor. And then after I added the value, then I increase my count on my counter by 1. And then at the end, after I iterated over all of my neighbors, I can divide the average, I'm sorry, by the number of neighbors. SBMLDN is the reference to the SBML model that we called DN and that we loaded into each of our cells. Okay, so in the previous part, we defined the antimony string and we loaded into all of our cells. And when we loaded it into all of our cells, we call it DN. So each of our cells has a DN model. And then what we're doing here is a little bit complicated, but the idea again is that we are the idea here is that we are basically averaging the delta value of the cell neighbors. And counter is just I'm doing counter to know how many neighbors that cell has. So every time I visit a neighbor, I store the value of that neighbor's delta, and then I increase the counter such that I can then average the delta over the number of neighbors that I have or that I visited. Okay. Right. So I added if counter because let's say the cell has no neighbors. I want to avoid a division by 0. So I added the if counter just in case my cell has no neighbors, my counter is 0. So I do not, the average remains, that stays 0 and I do not divide by 0. Ying, that's an excellent question. I always have, TJ or Julian are probably better to answer that question. What range does the neighbor function iterates over? And I'm pretty sure we've tried to figure this out by.
What do you mean by the neighbor function? There's a setting for that.
The for neighbor loop is what we mean. When we get the self get neighbor data list, whether that includes the corner neighbor. It's basically asking what order the neighbor visits over.
Right. It should be set by the lattice definition.
Right. But I am actually not 100% sure about that. I think it should be, but I'm not 100%. That's why I'm passing on the question. Let me launch the poll just to make sure that everyone is with me here. It's also important because this is actually the key part of the model. We haven't done anything different from what we've done in the previous exercise until this part, whereas where we are trying to connect the individual delta notch model in a single cell with some properties of their neighbors in the CompuCell simulation.
Could you explain a little bit about the for loop that you write here? And you said neighbor, comma, common surface area. These two, they are two variables there in the for loop, right?
Right. So when Python allows you to iterate over two different variables at the same time, as long as you're iterating, let's say, over something that can assign values to both of them. And then this cell neighbor data list gives you the reference to the neighbor. That's what we're calling neighbor here. It's the reference to the neighbor. And common surface area gives you how many pixels they have in common. How many pixels of your cell are in contact with the neighbor.
Okay, so
does that answer your question?
Yeah. So, but you didn't use common surface area in the loop, right?
Right, I did not. I didn't. I mean, that's also something we could potentially do. We could scale the delta value by the common surface area. That is a little bit of a different way of connecting it, a different way of thinking about how to connect the models, right? In this case, I just took the raw number of delta, regardless of how much common surface area they have. But in principle, right? Since delta is a cell surface receptor, it would make sense to scale my delta, how much delta I read off from my neighbor, by how much common surface area I have with that neighbor. So that's an excellent question.
So if you delete the common surface area from there, does it going to have an issue?
I don't think it works because the get cell neighbor data list is
two outputs, right?
Right, it gives you two objects.
Okay, got it.
Right, so that's why Paulo is explaining. The function returns a tuple, so you need to iterate through it with a tuple as well, with a tuple as well. Cool. So again, the idea here is this is an important part of this code, because it's a part that is allowing me to connect the delta notch in one cell with the delta notch in a different cell. So now that we have calculated this D average, all we need to do is to update in the cell the value of what we call D bar based on the average delta over the neighbors, right? So I am already iterating over my cell. So all I need to do is to now reference the delta notch model inside my cell and override the value of what I call D bar to be the average. Okay. That part is important as well, because in the first part, I just calculated the average delta of the neighbors. And in this part now, I'm overriding the D bar parameter inside the delta notch model with that value that I calculated in the upper part. Okay. So now, so Giuliano is pointing out that even with neighbor order or the pixel range 2, this function is always returning first order neighbors, which means that the ones that are in contact with the edges of the cell, not with the corners. Yeah, I wrote it a little bit different in this code than in the code that I'm showing in the as an example. Just a matter of writing it 2 times, but yeah, that is equivalent. That code that I had was just equivalent to doing this. It's just it is equivalent. Okay, so now now we have actually finished building our delta notch simulation. But if we run our simulation, even if we have coupled everything, there's really nothing that displays that. Oh, sorry, this is important. I forgot. We need to enable the neighbor tracker in the XML for this simulation to run. So to enable the neighbor tracker, we need to come to the XML model, do CC3D ML, plugins, and neighbor tracker. That's it. That is an additional plugin that I forgot we had to initialize at the beginning when we were initializing the simulation. But again, if we forget to initialize a particular plugin that might be necessary later, we can always come to the XML and add it. So yeah, if we run our simulation now, the model is actually solving the simulation is actually solving the delta notch models, but we have no particular way of visualizing how it's been resolved. So to be able to visualize it, we will do what we did in the previous example, where we added a auxiliary field to be able to see it, to see the values of delta and notch, in this particular case of delta and notch in each of our cells. I'm gonna go out a little bit quick over this because we've done it already a couple of times. But just as a reminder, in the init function, we go to CC3D Python, extra fields automatic tracking, and we need 2 of those. Right? And then let's call one delta. And call the other one notch. And for the attributes, we'll just call them D and N. And then as Paulie pointed out in the previous example, always good to initialize the attributes in the start function. So we will initialize the attributes D and N for every one of our cells. And then when we iterate over our cells, what we will do is that we will update how to add cell tracking. So CC3D Python, extra field automatic tracking, and that is done in the init function. Right, and we need two of those, one for delta, one for notch. Then we've created these two attributes in the cell dictionary, D and N. And then now we have to update those attributes in the step function. I'm sorry about that. In the step function, when we iterate over our cells, then we're just gonna update the values of delta and notch by referencing the values inside each cell. And again, to reference those, we can always come to CC3D Python, SBML solvers, get SBML value, get SBML value for SBML in a specific cell, right? So we need that reference. And we call it D in the SBML model. And N in the SBML model. And I'll launch the poll just to make sure that everyone is with me. Oh yeah, I'm sorry. It's DN model. Good catch. It's not model name, it's DN model, right? Good catch.
Sorry, maybe you mentioned it before, but maybe I didn't get it. Here we use cell.spml. So does it related to the SPML markup? I mean, that language, or is it Antimony that we are using here?
So the reason it's called cell.sbml is it's also explained here in the comments. I didn't go into it and I'm sorry, but the reason it's because even though we declared the model as a antimony string, CompuCell translates it into an SBML model and then into an SBML model. And then we, after loading it, we can just reference it as an SBML model. So that's why we reference it using that SBML.
Okay, you mean that when we write it as an antimony, then automatically it converts it to the SPML, then
yes,
we need to call that function because of
right.
that. Okay, thank you.
Yep. And I'm actually giving time. The reason I'm paused is because I'm giving time for the people who are still working on it. So now, if we come back to, just to make sure, because I had to take off my AirPods, I ran out of battery. Everyone hears me okay?
Yeah.
Okay, good. So now if we rerun our simulation, there was a typo. Somewhere I typed dig instead of dig. Just to make sure that I didn't repeat the error anywhere else. Yeah, I did. It's just a typo on my part. So now I should be able to see delta and notch. From my previous simulation, in my previous simulation, we fixed the values of D and N or the variables that we were using. So I have to unfix them here now. If I want to be able to see the actual patterning. And again, to fix the color bars, I always do this in the preferences. And the last thing, so now if I run it, if I run it, nothing is going to happen because I've actually not initialized any values of delta and notch in our cells. So even though our cells have the capability of implementing the delta notch model, we haven't given them any delta or any notch. So what we will do to initialize them is that in the start function, there's two ways we can go about it. We can initialize them in the antimony string, in which case all of the cells are going to start with the same values. But what I really want to do is I want to initialize each cell with its own value of delta and notch. And what I'll do is that in the start function, when I'm iterating over my cells, I'm gonna sample from a uniform distribution. I have it here. To initialize each cell with a random number or state for the delta and notch variables. And note that to do that, I need to import an additional library that is not immediately available in CompuCell, and this is a Python library, and it's called NumPy. Probably some of you are already familiar with it. Probably some of you know it even better than I do. But yeah, what we will do is that we will import this NumPy library to be able to sample from this uniform distribution to start our delta and notch variables. So all I did is I imported the library NumPy as NP. And then in when I'm initializing my cells, I assign a random number, a random value between 0.9 and 1 to the notch and delta in each of the cells. And I've just realized that if I run this model, it's gonna crash. And the reason it's gonna crash is because I am referencing the SBML model before loading it to the cells. So I need to move this, the loading function, before I actually assign the values, the initial values for each cell. Okay, so just to make sure, I'm gonna repeat what I did here. So up to here, we had initialized the SBML model in all of our cells, but we haven't initialized any values of delta and notch. What we're doing now is that we are initializing delta and notch to be a random number between 0.9 and 1 in each of our cells. And to do that, I'm using this NumPy library that I just imported. And assigning a random number between 0.9 and 1 to initialize those values. Okay, I'm waiting for, there's a couple of people still working on it. I'm just waiting on people to catch up. For those of you who have already caught up with me, I would recommend you to run the model yourself and then write on the chat, what do you see when you display delta and notch? So AJ, are you seeing some sort of pattern? Good. So I'm gonna run the simulation myself. And see what we get. So I do see some type of patterning, but it's kind of incomplete here. Let's launch it again. That's delta. And that's notch. So it looks more like this, probably. That was just a matter of what the initial condition, random initial conditions were. But now you can see that that delta, that those cells that are high in delta, which are the red ones, are also low in notch. And the cells that are high in notch are also low in delta. And that results in a patterning. If you have a little bit of a different, I would recommend that you go back to the equation. When you wrote the delta, the antimony string, just make sure that the antimony string resembles this one, which matches the equations, and that you're calculating the average delta of the neighbors correctly. Okay, good. Okay, how can we say this as a video? Excellent question. I don't know how this works in NanoHub, but in a local version, you would come to your configuration settings, output, and here you have the option to save the images every number of Monte Carlo steps. So at least locally, when I do it, this stores images of at the frequency that I specify here, saves PNGs of the simulation domain as it's being as the simulation goes by. And then once I have all of those images, then we usually take all of those images and use external software to turn them into movies. Okay, again, if it was a bit of a complicated exercise, so if your simulation does not matches these results, there's I would recommend you to go back to the code and make sure that there are no typos in your model string, no typos in when you're calculating your delta average. And there is a I've uploaded exercise 41413 to as an exercise so you can reference it back to make sure that you have no typos. So the last thing I want to show, and I'm just going to show really quick, is how we can have access to some of these models that we were looking at. There is a so okay, the second part of this exercise is to add staging panels. I'm not exactly sure. I'm actually not sure what I should do next. So Tarim, please make sure that your visualization settings are correct. So make sure that when you come to your CC3D preferences and you come to your fields, that none of the values are fixed. If they are fixed, you might not actually be able to see the fluctuation, the changes in delta notch. So that's a third reason you might not be seeing the patterning. So if the code is the same and it's working, make sure that your visualization are is working, and this is what you should expect to see. So in the next exercise, this is not complicated because it builds on this one. I will show how to add staging panels that allows us to modify the parameters of the delta notch on the fly. And on the fly, I mean as the simulation is running. And the reason we might want to do that is because, as James explained earlier, when you build these models, you might build them in explanatory, at an explanatory stage. And in that explanatory stage, you might want to explore how changes in the parameter values affect your simulation. So that's what we're gonna do now. So just keep work. If your simulation is working, we'll continue working on that one. If not, you can always start working from the demo exercise 413, and then we can work together from that one to put the steering panels, to add the steering panels. Oh, James had a good point. Once you've, for those of you interested in saving images, you have to click on save image every nth Monte Carlo steps, and you specify the frequency you want to save them. And once you do that, you hit on the picture on the camera icon in all of the windows that you want to save. And then, at least in my local version, every time I run CompuCell, that creates a directory in the CC3D workspace corresponding to that simulation. And then the pictures of the simulation are saved into that folder. Okay, so coming back to adding steering panels, the first thing we need to do to add a steering panel is to define this add steering panel function inside the step aware. So what we will do is that we will define the add steering panel function, and that's below all of the other functions in our step aware class. It takes self as an input. And then we need to specify which of the parameters, or what are the names and values and ranges of the parameters that we're gonna include in the steering panel. And the way we do that is using this add steering param function that takes the name of the parameter, and we're gonna call it A, because delta notch has a parameter called A. The value is the reference value that the simulation is gonna start with, or the steering panel is gonna start with. We're gonna set that one to 0.1. The minimum value of we're gonna let this parameter change. It's gonna be called min val. Gonna set that one to 0. And the maximum value, we're gonna let that parameter change. Max val, we're gonna set that one to 0.02. The decimal precision of the steering, in this particular case, is gonna be 3 decimal spaces. And then the name of the widget type that we want to use, and that we have different widgets. We have pull down menus, we have sliders. In this case, we're going to use a slider. So this creates a steering panel for parameter A. And I'll use this as a template to create another steering panel for parameter B. So again, I changed the name of the parameter B, and in this case, it's gonna go the default value is 100, the minimum value is 0, the maximum value is 200, and the decimal precision is 0, which means that the increases are integers from 0 to 200, and also a slider. Now, if I rerun my simulation, I should be able to get a steering panel that allows me to change the values of parameter A and parameter B. And just notice that parameter B, the maximum value is 100, and I wanted it to be 200. I'm gonna launch the poll to make sure that people are with me. All I did was I added this code to my previous simulation to be able to generate the steering panels. So if you can notice that even though I created the steering panels are not changed as I change the values of the steering panels. The steering panels are not really changing anything in my simulation. And this is because even though I created the panel, I haven't specified how the changes in this panel changes any value of my simulation. So that's what I need to do next. Next, I need to specify what the value of in the steering panel corresponds to in my simulation. To do that, I need to add a second function called process steering panel data, takes self as an input. And then to get the value, what value I'm passing from the steering panel, I need this function, self get steering param, which basically is reading the value that I'm passing from the panel in A. And in B. And I'm gonna store those values in these variables A and B. And now that I have the value, all I need to do is to update the values of the parameter inside the SBML models in my cells. I have multiple options. I could create whatever conditions I want, and I could choose whatever cells I want to change that parameter in, but I'm gonna just change it in, I'm gonna change it in all of my cells at the same time. What cells you change your parameters in, of course, are gonna depend on the particular program of interest. Here, just for simplicity, I'm just gonna change it in all of my cells. And this is just the same syntax that we use to update the D bar, but in this case, we're gonna update A and we're gonna update B. Okay, I'm gonna relaunch the poll. So now if I run my simulation, gonna let it settle until my cells are either high in notch or high in delta. And now I can start exploring how changing the parameters of might change the values in my simulation. It's not doing anything. Let's see. Okay, let's make sure that I'm doing it correctly. I do have the parameters A and B. Yeah. Not exactly sure what's happening because it was working earlier, but I have a working version that shows how this is supposed to look like. Is exercise 414. And again, if I run my simulation, delta, and let it settle. Now we can change the parameters. Let's see how that changes our model. So if I set A to 0, I lose my patterning. And then the more I lower beta, the higher everyone is in delta. This is of course related to what A and beta correspond to in the model, in the OD model that I showed earlier. But I can probably, by exploring this, I am basically exploring how the magnitude of the activation and inhibition of delta and notch, what the effect of changing the magnitude of the inhibition and activation are in the pattern formation. That allows me to explore certain biological hypotheses about, okay, if this is my the steady state of my system, what happens if I perturb it in this direction, if I increase the inhibition of notch on delta, or if I increase the activation of delta and notch. Oh, I'm sorry. Okay. So I'm gonna stop this simulation, this exercise here. Again, everyone for people who want to try it at home, it's exercise 414. So the last thing I wanted to show in this module was how we could look up for some of these SBML models. We've shown previously in the previous simulation, in the last couple of days, we showed how to make our cells divide in CompuCell, and we set a very simple condition for our cells to divide. We asked the question, have our cells double their size? And if they had, then we will trigger cell division. But in reality, there are multiple checkpoints that cell has to go through, and to be able to go through the full cell cycle and divide. And there are markers that indicate that the cell has successfully gone through some of these checkpoints. And the cycling, the proteins called cyclins are regulators of the cell cycle, and they're also markers that the cell has gone from one phase to another in their cell cycle. There are mathematical models of the cell cycle that we could use to incorporate into CompuCell, and then make for example, if we wanted to explore more complicated models of cell division, we could potentially include the cell cycle models into our cells, and then base the conditions of the cell division on those internal cell cycle models. And there is this particular model that was published by Tyson and Novak in 2001 that has a bunch of differential equations. But from those two are really important because they show the oscillations that we would need to characterize the cell cycle. The details are complicated, and we're not gonna look into the details of that particular model. But what is important, what I did wanted to show is how we can have access and how we can look up for this model. And there is a repository of models called biomodels, where we can go to and we can look up for SBML models of interest if we wanted to look for more intracellular or subcellular models. We just type the type of model that we're looking at, or if we know what the identifier of the model is, we could also look for it. In this particular case, we were looking for the cell cycle regulation model, which has this identifier. Once we have identified the model, we can go into the model and the files and look for the XML file that contains the model as an SBML. And then what we do, once we have that SBML, we can import it into our CompuCell simulation and load it into our cells. Let's say we have a single cell that we want to load this SBML model into. All we need to do is we have this functionality, add SBMLs to cell, that allow us to load the SBML model directly, even if we didn't specify it as an antimony string. Let's say we have an SBML model, our SBML file that has the model. Then we can load it directly as an SBML by referencing the model file. So far, we have used a model string, but in this particular example, since we have it as an additional file, we can load it into our cells. Then we can display whatever variables of that model we want using the auxiliary fields. And if we load it in a single cell and run it, what we will see is that the values of the cyclin B are gonna just increase and decrease continuously. So it's gonna oscillate from it's gonna oscillate from going up and down. And since we know that the cycling B is going up and down, then we can establish a condition for cell division. So for example, in this condition, we say that if cycling B has dropped below a threshold, the 0.5 threshold, and this is where we have established the condition. Remember, we do this in the mitosis step already. Then we trigger cell division on that cell. So basically we have put the cell cycle model inside each cell. We're measuring the value of the cyclin B. If cyclin B drops below 0.5, then we divide that cell. And if we execute that simulation, what we will see is that the cyclin B starts increasing, and once it decreases, the cell divides. And then now we have two cells with the same amount of cyclin B. And when the cyclin B in that cells increase, when the cyclin B decreases, then those cells divide as well, and so on until they fill the whole simulation. And this example is available for you in exercise 416. Of course, there's interesting questions that you could ask about this model. What happens, how we could implement different values of cyclin B in each cell, because we know that the cells do not go, even cells that are seated at the same time do not go over the cell cycle all at the same time. So this is an approximation, but we could, those kind of things you could potentially do, but by rewriting the values of the cyclin B inside each model, inside each cell. Okay, and that's the end of my simulation, and I'm gonna thank you for listening and for sticking with me, and I'm gonna hand back to James now. So there is a question, and I'm pretty sure TJ, I actually have that question too. Yeah, thank you for asking it. Does CompuCell support integrating the SBMLs with a stochastic simulator?
Yeah, this is a great question and a very new feature of CompuCell. So the simulator that we use, which is the same as what's used in Tellurium, actually has a whole array of integrators that are available that you can select as inputs. And one of those is a Gillespie solver. So if I remember correctly, there are 5 available in libRoadrunner, which is the simulator, and 4 of them are deterministic, and 1 is the Gillespie algorithm. And the way that you can select that is that in each of these functions that are available for creating antimony or SBML models and attaching them to cells, each of them, they all have an optional keyword argument called integrator. Remember correctly? And that keyword argument integrator, you can pass to that the names of the different solvers that are available, and one of them is Gillespie. So I'll type this into the chat. You can enter that. And I'm frantically searching through our manual to find you the reference on that, just for your own reference. And but of course I can't find it off the top of my head. So yeah, if you want to shoot me an email and I'll send you a link. But yeah, that's available for Antimony, SBML, and CellML models. And then in one of the modules that we'll cover tomorrow, that's also available for our Boolean network model simulator, which is has MaBoSS is the backend on that. That also uses a Gillespie integrator. Tarun asks, is the integrator available in CC3D Python? Yes. So when you type, say, cell.sbml and then give the name of a particular model, that actually gives you the entire solver, which has the integrator attached to it. So you can feasibly write cell.sbml. Whatever the name of your model is, and then .integrator, and that is the actual integrator that's being used.
Hi there, I have a question.
Please.
So I wanted to go back to the thing about in the notch delta model about iterating over the neighbors and the thing where it said common surface area. What I
I'll turn
wanted
it
to,
back to Josh then. Okay.
Yeah.
Josh, you want a screen share?
Yeah, 1 second. Let me get that part. Here.
Yeah. So I thought that's what I didn't understand is whether, in fact, as it's been written, the program, it is actually using that feature, or some additional code would have had to be put in to take advantage of that feature. Because it seems, from a physiological point of view, that is a good feature, because if two cells are in contact with each other, it's only the delta that is on the surface of the adjacent cell, which is in contact, that is relevant to the amount of notch signaling that a cell receives. So rather than counting all the delta that is in the neighboring cell, just what's in the contact area should be important. So is that what the way it's written, it's doing it, or does something additional need to be written for that to happen?
So yeah, so when the for loop, it's iterating over right, the get cell neighbor data list gives you that information, so it's already available to you. Whether you decide to use it, as I mentioned, it's up to the user, right? So for example, we could have divided where we have df there, we could have divided by that variable common surface area. Because that would make sense, right? And that's what I was explaining earlier. It would make sense that how much of the delta it's contributing to the signal depends on what the common surface area is between the cells, because these are cell surface receptor, right?
Correct.
So it would make sense to divide the common surface area by that, at least in this particular example. But it's available to you because the for loop is already iterating both over the neighbor, the reference of the neighbor, and what is the common surface area between your cell and the neighbor. So yeah, it's available to you.
Okay, thanks.
Mm-hmm.
Questions, comments? I realize that iterator over neighbors is sometimes confusing because people aren't used to seeing an iterator that returns a tuple, and so it looks funny to have two arguments in the loop, but it's quite a powerful feature when you use it. there were some questions in the chat about converting them, having trouble with one of the things one does have to be careful about is depending on where you've installed CompuCell. If you run a demo, you need to make sure that you have right access to wherever CompuCell is going to try to put its output. And so that can be a bit of a problem. Sometimes it can be confusing for people. So you do have to remember to set your target directories to be something that you have right permission for. Tomorrow, we're going to start out with what would have been the last talk today, which is going to be on MaBoSS integration of CompuCell to do stochastic Boolean network models. Then we'll have 2 talks on links to do convergent extension and epithelial tissue folding, which are going to be very relevant to the hackathon ideas. And then we'll have a final talk on parameter scans and data export and parameter files and model building. And so I hope that that works. If people are still having trouble running the Antimony integration with the CompuCell, please do contact us, and we'll do our best to get a patch out for CompuCell. TJ, do you have any suggestions, final suggestions for what to do?
I mean, we've tried a couple of solutions that we've had intermittent success with, but I'm still getting reports from people who are not finding those solutions successful. So the surefire way that we do know of that's available to everyone is to migrate over to the NanoHub tool. So, you know, in the module that we do on MaBoSS, for example, there's a bit of additional installation work to do MaBoSS and CompuCell locally. So in that module, I'm going to ask everyone to migrate over to the NanoHub tool, if only for that module. But if just don't want to waste time on trying solutions and things like that, then I would strongly urge you to use the NanoHub tool. And then of course, use the functionality to migrate your simulations to and from their servers in your local machine.
Josh, there was a question. AJ, do you still have a question, another one, or is that for the one you already asked?
Sorry, I should put my hand down.
No, it's just I wanted to make sure. So there was a question in the chat. Josh, I don't know if you want to discuss it. There was a question about, it says if you have more than one compartment of a cell, how can the products from one compartment link to the other compartments?
So, so the way we define compartments is by creating, new cell types, and then we basically tied the 2 different cell types, and then we call that a compartmental cell. And it would follow the same principle as we were doing before, where you load, let's say, one antimony model in a particular cell type, will correspond to loading it into a compartment of a cell, and then you can link it the same way we've done it so far, linking between different cells, but instead linking it inside a single cell. I don't know if that answered your question. So basically, yeah. So basically, it follows the same principle as linking between different cells, because in principle you treat the 2 compartments as if they were different cell, what we call cell objects.
That would be a good exercise to try. You could try.
That's what I was thinking. Never tried it before. It would be an excellent demo to have.
Any other questions? Okay, so I hope I hope it works. If it doesn't work the way you expect, we'll find a workaround for it. Any other comments, suggestions? I know it's been a long day. I appreciate people's patience with the issue with the Windows installation. Great. Okay, well, I guess then we're done for today. If the instructors could stay on for a minute, that would be great. If there are additional questions, well, you're welcome to ask them.